Good morning, everyone. It is my pleasure to welcome you all to the technical session on crop science and emerging technologies for sustainable agriculture in First International Symposium on Agrotechnology and Rural Sciences 2025. I am honored to introduce and invite our esteemed panel chair, Emeritus Professor S. Subasinghe, Professor in Crop Science, Faculty of Agriculture, University of Ruhuna. Professor Subasinghe is a distinguished academic whose contributions have significantly shaped the field of agricultural sciences in Sri Lanka and beyond. And also, he served as one of the former director of the UCIARS. Then, I would like to invite our panel members, Mrs. S. Navaratna, Senior Lecturer, Department of Food Technology, UCIARS, and Mr. Hasanaka Rohanadira, Department of Agrotechnology, UCIARS. Dear Sir, Professor Subasinghe, I warmly welcome you to chair the session and proceed. Over to you, sir. Thank you.
Good morning, everyone. Can you hear me? Can you hear me, please?
Yes,
Can
sir,
you
we
hear
can
me?
hear you. Yes, sir, we can hear you.
Okay, good morning, everyone, and welcome to the technical session 1. And thank you very much for inviting me to chair this session. There are 10 presenters. I directly am the go to the presentations. The first presentation on optimizing cauliflower growth and yield in hydroponic systems, a comparative study of A-shape and U-shape hydroponic structures by D.U.N. Madhusankar and K.A.N. Sevan. I think Ms. Madhusankar, Mr. Madhusankar is going to present.
Hi everyone, and thank you for being here today. My name is Nirunda Masanka and I am honored to present our research title Optimizing Cauliflower Growth and Yield in Hydroponic Systems, a comparative study of A-shape and U-shape hydroponic structures. This is the outline of presentation. Cauliflower is versatile crop grown across Sri Lanka's climatic zones, but traditional cultivation faces challenges like extreme temperatures, water scarcity, and pests. Hydroponics offers solutions enabling year-round production with efficient water use and controlled environments. Despite hydroponics' potential, two critical gaps exist. First, few studies examine cauliflower in hydroponics. Second, no comparative analysis of A-shape and U-shape structures for this crop available. Our objectives were clear. Compare growth metrics like plant, height and leaf count, and evaluate yield differences in head weight and diameter between the two structures. By addressing these gaps, we aim to empower farmers with data-driven insights for sustainable cultivation. Our experiment used a randomized complete block design with 3 replicates per structure. We grew snowball Y cauliflower in coir dust and clay pellets with nutrient solution monitored to pH and light intensity at 3000 lux. Growth parameters, plant height, canopy diameter were measured weekly, while lead metrics like head weight were recorded at harvest. Statistical analysis via ANOVA and Turkey's HSD test ensured robust results. The A-shape structure outperformed the U-shape with plants reaching 46.16 centimeter, 14.94% increase in height. Similarly, canopy diameter was longer in a shape 51.2 centimeters. Differences like stem from a A-shape's superior light distribution, reducing shading and promoting vertical growth, a finding aligned with previous studies on light role in photosynthesis. Yield metrics further validated the A-shape's advantage. Total biomass yield in a A-shape 1.88 kilograms per square meter was 51.61% higher than the U-shapes 1.24 kilogram per square meter. Head perimeter, a proxy for curd quality, was also longer in the A shape, 36.5 centimeter, notably higher fertilizer rates, 2 grams plant per day, synergized with the A-shapes design boosting yields. This underscores the interplay between structures and nutrient management. Why did the A-shape excel? Two factors, light and stress reduction. The A-shape's geometry ensures uniform light exposure, critical for canopy formation and curd development. Meanwhile, reducing shading minimize environmental stress combined with optimal nutrients. This created an ideal micro environment. Simply put, the A shape is not just a structure, it's a growth accelerator. In conclusion, the shape hydroponic system significantly enhances gold power growth and yield compared to the U shape, thanks to better light distribution and nutrient synergy. For tropical climates like Sri Lanka, this offers sustainable pathway to year-round production. Future research could explore other basicas or scale-up trials for commercial adoption. Thank you for your attention. I had be happy to take any questions. Thank you.
Thank you very much, Mr. Madhusankar. Now the paper is open for discussion. Any questions or comment, please? I have, sorry.
Yeah, Ms. Madhushankar, it's a nice presentation. So I have one clarification only that based on your research, you have checked that two shapes, structural shapes, A and U. So have you make any cost analysis for that the material cost or make that the structures there? Mr. Madhushankar?
Madhusankar, can you hear us? You on that mute? Yeah, okay. Can you hear the? Madhusankar? What's wrong? He is there, but I don't know. Some issues on network maybe. Madhusankar, can you hear us? Weak internet, ni? Madhusankar, can you answer Mr. Vanadira's questions? No, again some. Madhusankar, you are again mute.
Hello, Professor.
Yeah, can you
hear me?
Yeah, we can hear now. Can you answer the question raised by Mr. Vanadira? Mr. Vanadira?
Please, can you again question me?
Can you ask again, please?
Yeah, sure. Thank you. Ms. Madhushankar, based on your research, you have conducted the research with that two shapes, A and U. Have you
Yes.
make any, for the just clarification, have you made any cost analysis for this production cost for A and U, or they are the same in cost or something?
Yes, thank you for the question. We analyzed the both structures, and they are compared to cost of A is compared to price is high for the there are some structures are additionally at than C structure.
So it means that based on your result, that A-shape is the most efficient system for that polyplan. So anyway,
Yes,
that
yes,
their
sir.
production, that the structure cost is higher. Okay, thank you.
Yes, sir.
Shankar, I have a
Yes, sir.
question and clarification.
Okay,
Have you done
Professor.
this experiment inside the protected house?
Yes, Professor. It's done in a greenhouse structure.
Right, right. Then why you that your experimental design was RCBD? Why you use RCBD?
It's because they have different they are significant of that result.
Not clear.
Hello, dear Professor, can you hear me?
Yeah, yeah. Yes, you can explain. Shankar. What was the
Because
reason for
they
RCBD?
are. Can you hear, Professor? Yes.
Yeah, yeah.
Because Professor, we use multiple get the parameters, and we need to get significant of that results. That's why we use that Turkish that model.
Generally, inside the protectors, no, and the difference of the environment over everything. But the one thing is, the other thing is, you use two structure types and two fertilizer rates, right? So
Yes,
then, have you arranged in factorial design or just RCBD?
just RCPD.
No factorial.
Yes, sir.
Then, what is the basis of selecting this 1 gram and 2 gram per plant?
Because that 1 gram per plant is a normal optimum rate for the cauliflower cultivation, but we need to get optimum nutrient requirement for the cauliflower cultivations. That's why we use the 1 gram per plant and 2 gram per plant nutrient rates for the evaluation.
Optimum gram means the recommendation, Department of Agriculture recommendation or what?
It's got from research the previous have done in Sri Lanka. Yes.
1 gram.
Yes.
What is the farmer's practice?
Actually, this one is we are using hydroponic systems, but normally in Sri Lanka tropical in upcountry regions, normally they are cultivation in the soil.
In your abstract, you didn't, nothing mentioned about experimental design. And no, that when your presentation also not much explain about the influence of fertilizer rate, 1 gram and 2 gram, rather than just mention 2 gram per plant to increase the head weight. And that is the most important part. That yield. That should be more elaborated. But in your abstract also nothing mentioned on that, except in the conclusion you just mentioned alpha fertilizer 2 gram per plant increase the head yield. Right, those are my comments. Right, any other questions or comment? If there is no any more questions or comment, thank you very much, Mr. Madhusankar. So shall we move to the
Thank you.
Yeah.
Thank
Shall we move
you,
to the
Professor.
second presentation? by LGC Hansika, G Navendra Kumar, and NRN Silva. I think Hansika, Miss Hansika is going to present on comparative study of dairy sludge and municipal waste sludge with the normal compost on okra, growth and yield.
Good morning all of you. I'm Chamodi Hansika from University of Vaunia. Today I'm here to present about my research project which I conducted at Horticulture Crop Research and Development Institute, Gannoruva. The title of my research is Comparative Study of Dairy Sludge and Municipal Waste Sludge with the Normal Compost on Okra Growth and Yield. Let's move to the introduction. Chemical fertilizers can cause a lot of negative environmental impacts like water contamination, nutrient runoff, algal blooms, soil degradation, etc. So there has been a shift toward using organic fertilizers. Some recent studies suggest sludge can be used as organic fertilizer, so I wanted to explore that idea further. Actually, sludge has more nutrients like NPK and it enhances soil fertility, reduces dependency on synthetic fertilizers, promoting sustainable agriculture. Since my research period was limited to just 4 months, I needed a crop with a short growth cycle. So I chose okra because it has 2 months growth cycle. My research aim is determine the possibility to use of sludge from a dairy industry and municipal wastewater treatment plant as a fertilizer for okra growth and yield. Objectives are to determine the composition of 2 types of sludge, to evaluate the effects of 2 types of sludge and their combinations with compost on the growth, yield and nutrient uptake of okra, and to explore its potential reuse as fertilizer for okra growth and yield. Let's move to the methodology. Take dairy sludge, municipal sludge and normal compost. Analyze the composition of them. Carry out the pot experiment with okra. Determine these kind of parameters and compare the results with Department of Agriculture recommendation. I collected dairy sludge from Milko Private Limited, Digana, municipal waste sludge from Kandy City Wastewater Treatment Plant, normal compost and soil from Horticulture Crop Research and Development Institute. This is the chemical parameters I checked in the laboratory. Plant nutrition analyzer used to check leaf nitrogen and chlorophyll. I used 5 kilograms of pots and filled with 4,900 gram of soil and 100 gram of organic material based on this calculation, and I used MI5 okra variety. I did 6 treatments with 4 applications. I recognized some pests and diseases also. These are the parameters which I measured in the field. I used Minitab software for the data analysis part. One-way ANOVA for parametric variables, Kruskal values for non-parametric data. Let's move to the results and discussion. You can see red color and green color values. Red color values means the parameters which satisfy DOA recommendation, and green color means the parameters which not satisfy DOA recommendation. Here also the plant height, stem circumference, number of leaves, number of buds shows significant differences and others are not. Pot color. All okra pods were classified as 144B yellow-green group. Treatment 3 achieved the highest plant height and the highest stem circumference. I collected 32 okra pods from all treatments. Let's move to the conclusion. Treatment 3, which consists both municipal and dairy sludges, achieved the highest plant height and the stem circumference, increased buds and leaves. Treatment 4 only consists normal compost, shows the lowest plant height and stem circumference. The highest yield was in treatment 3 which consist both municipal sludge and dairy sludge. There are some limitations. Use of clay soil, cause waterlogging, unmeasured soil nitrogen, restricted root growth in 5 kg pots. Recommendations are use sandy loam soil and large pots in future studies. Conduct heavy metal analysis. These are my references. Thank you.
Okay, thank you very much, Ms. Hansiga. Now the paper is open for discussion. Any comments or questions, please?
Yes, thank you very much for your nice presentation. I have a small clarification. Miss Hansan, as a treatment, no? So, was it prepared by yourself in your research study place, or it's a commercial available compost did you use for your research?
I prepared, miss. Can you hear me?
Yes, but
You
not
prepared. So,
Yes, miss.
Ms. Hansiga, as you
Yes, sir.
recommendation you mentioned that in future studies we have to think about the heavy metal. That actually that is one of my questions to be raised here. That is the most important, one of the most important limitation in this use in municipal waste. If you analyze this in your that justification also you mentioned that rather than using NPK, this compost and all other that organic materials, there are some other important nutrients like micronutrients. So if you analyze and convince the influence of other nutrients also, I think much value on this paper. Then you have the different rates, two rates, right? Two rates of compost, two rates of municipal waste, two rates of what is that slurry, water treatment, the waste treatment plant, right? So then you just consider the factorial manner or individual treatment. You have 6 treatment now?
Yes, sir.
You just
With
consider
4
the factorial.
applications.
Sorry?
With
What
4
is the experimental
applications.
design? CRD? Just CRD?
CRD. Yes.
Not factorial.
No.
Then you mentioned here 40,000 kilogram per hectare of compost, the recommendation. Based on that, have you calculated this municipal waste and other that treatments?
Yes, sir.
Then you mentioned in your presentation as a yield parameter 32 okra head, just to number. I mean that sometimes if you little elaborate these yield parameters, sometimes the length may be different, weight may be different in okra, the head so forth. Then the number doesn't mean that much. But rather than just number, if you that mention the weight, I think much better because there's the market in commercially also important the weight, right? Fresh weight. Yeah, those are my comments.
Thank you.
May I get a clarification?
Yeah.
I'm Shashika from Faber Research Institute. Actually, it's nice presentation. Miss Hansika, in what percentage you mix the municipal waste and the sludge, dairy sludge in treatment 3?
50 gram, 50 gram, 50 gram. Both.
Means that
50
1 to
gram.
1%, 1 to 1 ratio.
Yeah.
That's all. Can I wind up the presentation? Thank you very much, Ms. Hansika.
Thank you
for your
very
presentation.
much, madam.
So then shall we move to the next presentation on effect of different seed coating and storage temperature on quality of maize seeds by GGA Sandamali, MGTL Priyantha, US Hera, and WCP Egodavat. I think Ms. Sandamali is going to present.
Good morning everyone, I am Manusha Sandamali from Faculty of Agriculture, Rajarat University of Sri Lanka. Today I am very pleased to present my research on effect of different seed coatings and storage temperature on quality of maize seeds. Seed security is very critical to ensure food security, and one of the effective way to enhance seed quality is through seed coating. And seed coating is the process of applying a thin protective layer of materials to the seed surface to improve germination, protect from pests and diseases, and to enhance storage life. So why seed coating technologies are needed in the seed industry? It is because this technique has gained popularity in the seed industry as an economical and effective method. As shown here, seed coating improves seed vigor and shelf life, protect from biotic stresses and facilitates easier handling, all while reducing environmental impact. So one of the major issues in maize cultivation is the use of poor quality seeds, which leads to reduced productivity, and seed deterioration during storage significantly affects germination and seedling growth. So this research is aimed to identify a practical and affordable solution to address this issue. So the broad objective was to evaluate the efficacy of seed coating in improving maize seed quality and storage life, and the specific objectives included assessing the effects of seed coatings and storage temperatures on both seed quality and storability. So the research was conducted at the Central Seed Laboratory of Gannoruwa, and we used a factorial completely randomized design with 3 factors. There were 7 coating formulations which were all replicated 4 times. So in this experiment, 6 coating formulations were tested along with an uncoated control. As the coating polymer, a ready-made polymer, and carboxymethyl cellulose were used. As the fungicide, homai, and as the insecticide, thiamethoxam were used. So these are the polymers used in the study. And in this slide, you can see the process of seed coating. First, the seed coating machine was turned on, and then the seeds were fed into the inlet of the machine, and the properly weighed chemical mixture was added to the liquid mixing vessel, and within 5 to 10 minutes, seed coating is completed, and the coated seeds were collected from the outlet of the machine. Here you can see the coated seed collected from the seed coating machine. So in the next step, seeds were dried using a seed dryer, and they were packaged in triple laminated aluminum foil packets, and the seeds were stored separately at 2 different temperatures, at ambient temperature and cold room temperature. So here you can see the final appearance of each 6 coating treatments. So seed samples were collected from each treatments at 4 intervals, as just after coating and after 1, 2, and 3 months of storage. At each sampling time, 3 main seed quality parameters were measured. First, the germination percentage was measured, and then the seedling length was measured according to ISTA guidelines, and finally the seedling vigor index was measured by multiplying the seed germination percentage by mean seedling length. So let's move on to the results and discussion. The germination percentage of all coatings decreased with storage period. However, the rate of decline was slower in coated treatments compared to control. In both Emma hybrid 4 and Emma hybrid 5, the highest germination percentage was recorded in C2 and C6, with both storage conditions showing similar results. Compared to coated seeds, in non-coated control, germination percentage dropped over 10%, reaching the lowest value. And in both maize hybrids, the highest seedling length was observed in C2 and C6, with both storage conditions giving similar results. All 6 coatings showed longer seedling than control, with the largest difference exceeding 3 centimeters. In both maize hybrids, the highest seedling vigor index was recorded in coatings C2 and C6. Stored under cold room temperature, followed by the ambient storage of same coating treatments. The lowest seedling vigor index was observed in non-coated control. In conclusion, the germination percentage of seed coating formulations remained unaffected compared to the uncoated control. All coating formulations showed seedling lengths and seedling vigor indexes higher than the non-coated seeds, and seeds and seedling quality were unaffected by the storage temperature. So among all coating formulations, coating 2 and coating 6 showed the highest performance, and they are recommended as effective seed film coatings for improving seed quality and enhancing storage life. So these are the references I used. Thank you for your kind cooperation.
Thank you very much, Ms. Sandamali. Now the paper is open for any questions or comments.
Okay, thank you, Ms. Sandamani, for your presentation. I have one clarification. Based on your results, you have mentioned that the no significant difference between that seed coating polymers. Is that correct? When it go for the storage conditions.
I'm sorry, sir. I didn't quite hear that question.
Yeah, we got that based on your results, when you explained that no any significant difference in a seed and seed quality at storage in coated seeds. Is that correct?
Yes, sir. All the coating formulations showed the better results than the uncoated control. Comparing to uncoated control, all of them were significant. No, not actually the highest result was in the coating 2 and coating 6.
Yeah, thank you. Got it. And how you measure that the seedling quality? What are the parameters you have utilized for?
For seedling quality, actually in the ISTA guidelines for finding a better seedling when getting the germination percentage, we have to take intact seedlings with at least 2 true leaves with a good root system. So likewise, there are lots of things to consider when considering a good seedling before taking the germination reading.
It means that morphological factors basically you have utilized, no?
Yes, sir.
Okay, thank you.
Thank you, sir.
Ms. Sandamali, you use this factorial randomized design.
Yes, sir.
Right? In factorial designs, generally we calculate and that in a statistical design, we are looking for this any interaction. Have you find any interaction on these factors?
Yes, sir. In interaction effect, actually, interaction was not significant. Actually, all the
factors.
We considered 3 factors: the coating treatment, temperature, and the variety. So they were
storage, variety,
and the film coating polymer, sir.
Levels also you use different levels of polymer, different rates.
Yes, sir.
Four factors.
Actually, only 2 types of polymers I used.
But no different rates of polymer. 6
No, sir.
distinct seed film protein formulation. What is
Sorry,
that?
sir.
6 different seed protein formulation.
Yes, sir.
Two hybrids, 2 storage temperature, 2
Yes.
varieties. Then any levels? 6 means 6 level means
Yes, in the 6 levels, actually,
then 4 factors. No.
I only considered the 3 factors, sir.
How can?
Yes.
I don't know. You consider the 4 factors and that in your experiment 4 factors, but you consider only 3 factors.
Yes, sir.
I don't know statistically. Yeah. Sanmal. Any reasons for why I'm that you are getting 4 factors, but just consider only the 3 factors?
Because the research was continued for 6 months, and I analyzed the data considering only 3 factors for my presentation, sir.
But you didn't consider about the different formulation.
Only I used a ready-made polymer and alternative for that as a low-cost method. I used carboxymethyl cellulose. So I wanted to measure the effect of carboxymethyl cellulose to use in Sri Lankan context as a low-cost method because the ready-made polymer is very expensive. So that is what I majorly want to evaluate. That is why I didn't consider the
That is true,
level.
but if so, you don't want to mention here that's different than the formulation, 6 formulation, right? Forget about 6 formulation. If you just consider only the 3 factors, right? But here your abstract also it was mentioned that 6 formulas and then 4 factors there. Then there may be some interactions also which we don't know if you want that consider. That's all. That my point.
Thank you, sir.
Any other comment, please? No more comment. Thank you, Sandamali.
Thank you, sir. Thank you so much.
Shall we move to the next presentation on influence of growing media and planting technique on growth of young budding rubber plants under nursery conditions by KKLS Chandrasena, SA Nakandala, UN Udayakumari, BS Bandhusekar. I think
The title of our research is Effect of Nursery Media and Planting Technique on Growth Performance in Rubber Nursery. The authors are myself, Lahuru Chandrasena, and Dr. Shashika Nakandala and Mrs. Udayakumari and Dr. Supun Bandhusekar. In the introduction, rubber tree, commonly known as para rubber, is belong to Apiaceae family. Rubber was introduced 1876 and planted first rubber tree in Henrathgoda Botanical Garden in Gampaha. Rubber tree traditionally grow on wet zone, dry zone and intermediate zone, and irrigation is therefore necessary for dry area and where along to dry spell are prevalent. With that, the research problem of a study was identify the impact of growing media and planting technique with irrigation on growth performance of rubber nursery plant as a feasible solution for water scarcity. The objective of this study was to evaluate the effectiveness of growing media and planting technique on growth performance in rubber nurseries with two specific objectives as to assess the effectiveness of growing media of rubber nursery plant on growth attributors, to evaluate the different planting techniques of rubber nursery plant on plant growth attributors. Experimental design. The experimental design was RCBD with two factors as different planting technique and different growing media. treatment were different planting techniques as planting on trench with coir pit, planting on trench without coir pit, and different growing media as T1 topsoil mixture, T2 topsoil mixture and coir pit 1:1 ratio, T3 topsoil mixture burn rice husk 1:1 ratio, T4 topsoil mixture coir pit burn rice husk 2:1 ratio, T5 topsoil mixture coir pit compost 2:1 ratio. Let's move to the methodology. About 5,000 rubber seedling plants were selected for the study. Nursery plants were arranged in nursery according to the experimental design with 5 replications, 10 treatments each containing 100 plants in double row system. Plants were irrigated with sprinkler irrigation system in dry period with the irrigation interval of once in 2 days. Plants were treated fertilizer and pesticide once in 2 weeks interval. Recommended agro-management practices were done throughout the nursery period. Plant were bud grafting using the clone RRIC 121 kept for 1 month. In nursery grafting were success. Success percentage was observed and recorded. Plant morphological and physiological parameters before and after bud grafting were recorded on stem diameter, plant height, leaf area, root growth, chlorophyll content, and stomatal conduction. Soil moisture content was measured during the experiment by using theta probe and moisture meter. Let us discuss the result obtained from this research study. More importantly, bud grafting success rate show higher success rate 85% or more plant potted with 1:1 soil and coir pit mixture both with and without coir trenches. Control plant and soil only show lower success rate under the both conditions. In plant growth measurement, these two graphs show the variation of treatment on plant stem diameter and plant height. T2 with coir and offer best condition for the stem diameter and stem height before bud grafting. And when sign growth, the change in diameter and height is significantly clonal character. Under morphological character, leaf area and root height were measured. The bar chart of root length present coir and soil mixture significantly increasing the taproot length across the different growing media when compared to the control treatment. In physiological measurement, plant chlorophyll content and stomatal conductance are not significantly variated with growing media and plant are not undergone any water stress condition. Soil moisture of treatment plant variated with the ratio of coir pit, compost and rice husk mixed with topsoil. The highest moisture retention was obtained T2 treatment and potted plant placed in coir pit trenches. With that, I would like to conclude my presentation as plant that are grown media mixed with coir pit and soil 1:1 ratio. T2 shown higher plant height, diameter, root density, followed by T3 and T4 treatment than the control treatment 1. The higher bud grafting success rate achieved with the most effectiveness growing media, coir pit and soil 1:1 ratio. T2 placing the polybag plant under the coir pit trenches. The results show that keeping polybag nursery plant on a coir pit trench conserve the more water can enhance the plant growth performance. The impact of a grow media with mixture, coir pit and different ratio and place coir pit trenches can be used to increase in water use efficiency as feasible solution for water scarcity when explaining the rubber dry area in Sri Lanka. Thank you.
Right. Thank you, Mr. Chandrasena. The presentation is open for discussion. Any comments or clarification?
Yes, thank you very much, Mr. Chandrasena, for a very clear presentation. So, yes, I have one clarification from you. According to your findings of this study, it's clearly suggested that coir-based media was enhancing the growth. with compared to the existing media what the farmers practicing currently, no? So have you identified any constraint or the obstacles that can be guessed while introducing this new finding to the farming community who are engaging with the rubber nurseries?
Well, topsoil depletion very high, then and poor structural and biological health. Then we are using claim this media. Um, it is a taproot and then this area is high growth using coepit and then that improve plant physiological and biological health and that improve plant plant growth and enhancing the soil health and climate adaptation to climate changes.
Let me, it has a less disadvantage or constraint, and we can easily identify it to the farming community. Then your finding, that means Firebase media can be introduced to the farming community.
Yes. Sorry,
Right,
hello?
okay. Yes, can.
Hello. Sorry.
Yeah.
Yes, we can hear you. That means Mr. Chandra Sena, can you suggest that Firebase media for the farming community, no?
Yes.
Yes, okay. Okay, thank you.
Any other comments, please? Ms. Shamsena, on clarification, what is the potting media presently used for young budding nurseries? What is the presently that used in RRIO? What is the recommendation?
RI121 clone.
No, I mean that protein mixture. What is the protein mixture they use?
May I comment, sir?
Yeah.
The protein media that we currently use consists of 50 gram of export HERP and 50 gram of compost and the topsoil. The issue is when we recommended to use topsoil, they never use some topsoils as the scarcity of finding topsoils. They use subsoil instead of the topsoil. That's why we try to test different types of potting media that we can increase their nutrient content and all the water absorption capacity. That's why Mr. Lahiru did his final year project with us and gets more reliable results on his study.
It is good, but if you can include and that whatever the existing treatments also, I think you can get better results to compare the existing one as well.
Yeah, yeah, topsoil in the case, we use that our recommended potting media.
Only topsoil?
Not the topsoil, that we mention it is as a topsoil, it is a recommended potting media. Additionally, we use other coir dust and the rice husk for the mixtures.
Yeah, I mean that you use for that control treatment only the soil mixture means what is the soil mixture? Only the topsoil for the control treatment?
Yeah.
But in your existing practice is soil mixture plus some rock phosphate and some other
Yes.
materials as well.
Yeah.
That is what I'm that if you can compare that one also better.
Right, sir. Okay, thank you,
Yeah.
sir.
Okay, any other comments?
Yeah, Mr. Chandrasena, one clarification only. Have you analyzed that NPK availability before you start your research and also just after the that research end? So have you analyzed the NPK?
Yes, yes, sir.
Okay. It means that what are the rates? It means that is up to that standards are tied with that or is below the standards.
Sorry.
In general, that NPK having some standard in when you go for the composting or something or mixing some with the nutrient with the soil, it should having some standards. So my question was, if you when you are analyzing, you can see that what are the rates of NPK in your soil mixtures. My question was, have you able to reach up to that level?
Hello.
Did you get my point, Mr. Chandrena? Anyway, one thing that have you checked that the NPK availability just after end of your research?
Yes, sir.
Okay, it's because that when you are comparing the morphological characters, that the NPK, that available nutrient make a that play a major role there. So that's why I asked you. Okay, thank you.
Thank you.
A small clarification. Your experiment was finished. Chandrasena.
Yes, sir.
The Indian experiment was factorial, right? 2 factors, correct?
Yes.
What are the 2 factors?
And growing media and planting technique.
Growing media and planting method.
Planting technique.
Planting technique, right? Then have you find any interaction effect in these 2 factors?
No, sir, that we could not find any interaction between two. That's why we separately
Okay.
mention it.
Okay. Thank you.
Thank you, sir.
Right. Thank you very much, Mr. Sena. Shall we move to the next presentation? Next
Thank you, sir.
presentation is identification of stigmasterol as a potential antiviral compound targeting ASFP DNA polymerase X, a comprehensive in silico analysis by S. Purni N. Jampati NMT Anupama WMPM Ulugalla N. Gamage KGA Samadhi D. Jayavardhana and L. Ranatunga. I think Miss N. Jampati N. Jampati is going to present.
Let me begin by highlighting the seriousness of African swine fever. For over 100 years, ASF has caused significant losses in animal populations, with millions of pigs affected globally. Despite the magnitude of the problem, there are currently no effective vaccines or drugs available to control this disease. ASF not only threatens the food security but also has a profound impact on global economy. Imagine if the nature itself holds the key to combating such a deadly virus. Today I'm here to share a fascinating journey into how stigmasterol, a plant-derived compound, could be the answer we have been searching for. So My research title is Identification of Stigmesterol as a Potential Antiviral Compound Targeting ASFV DNA Polymerase X. This is the content of my presentation. Moving on to the introduction, major outbreaks have occurred across the world, including significant incidents in Georgia in 2007 and China in 2018, highlighting its persistent and widespread threat. The disease led to internal bleeding and loss of appetite and inability to stand. It spread through contaminated vehicles, meat, and both domestic and wild pigs. ASF led to nearly 1.9 million animal loss worldwide. To address this, we used in silico analysis to study how potential compounds interact with the viral protein, aiming to identify effective antiviral agents. These are served as templates that guided our research approach. ACV is a large double-stranded DNA virus. ACV-POLX is one of base excision repair proteins. It is crucial for viral genome repair and is a promising target for antiviral drug development. Our main research objective was to identify active compounds in Geneva sylvestre plant and evaluate their antiviral potential against ASFV-POLX using in silico methods. Specifically, we assess drug likeness and binding affinities through molecular docking. This is the data analysis part we used. Let's dive into the methodology. When come to plant compound identification, we choose this plant, Gymnema sylvestre, using a thorough literature survey. It has been a cornerstone in a traditional medicine for centuries, offering a range of health benefits. Here we found 36 active compounds. Next phase is all about assessing whether our potential drug candidates are safe or not. In here, we applied Lipinski rule of 5. This filtered out compounds that are unlikely to be orally bioavailable. In here, ADME stands for A for absorption, D for distribution, and M for metabolism, and E for excretion. This step reduced our final amount to 19 compounds. When we come to protein and ligand preparation, we started up with crystal structure and we ended up with retaining only chain A, removing all the water molecules and adding polar hydrogen to optimize electrostatic interactions. Let's move on to the virtual screening. This step is crucial as it allows to narrow down effective compounds from a larger pool. As shown in the graph, we analyze the binding affinities. Binding affinity quantifies the strength of the interaction between a ligand and protein. With more minus value, it indicates strengthened binding affinities. Among all tested ligands, 14 compounds exceed this threshold, which highlighted in black bars. Now let's proceed with top 14 compounds. To confirm our results, we performed further docking using CB-Doc2, which provided additional validation. Our analysis identified stigmesterol as the compound with the highest binding affinity. This suggests a strong inhibitory activity against ASFV-POLX and potential as a lead antiviral agent. To advance our research, we need to conduct further studies using both in vivo and in vitro. These are the key references that supported our research. Thank you for your attention.
Okay, thank you very much, Ms. Jayampati. Paper is open for discussion. Actually, my expertise area is completely different, so I may not have any questions. But I'm interested on this medicinal herb since we did a lot of propagation studies on this Gymnema silvestre. So, any comments or questions, please?
Yes, one comment that Jam that as we know that the late month of the 2024 that the country was highly affected with African swine flu. So that's why your research may be important to find some kind of a laboratory analysis to find out any kind of chemicals or some extraction to avoid this kind of things happening to as well. So it's very important to go for, as you said, that in vitro and later on in vivo sessions to identify whether that the identified compounds are effective is for or not. So that's my comments. Thank you.
Any other comments or questions?
Thank you, sir.
Yeah. Any comments, please? Any other comments? Okay, thank you very much. Very interesting for me also, but it's beyond my that exercise field. But this medicinal plant, we did a lot of experiment on diabetes. Okay, thank you very much, Stanford. The next presentation is on efficacy of nutrient nanopractice infused slow release fertilizer based on sodium alignate and polyvinyl alcohol for chili plants. By I think Miss Tanya is going to present.
Good morning, all of you. The modern agriculture is working on introducing new generation of fertilizer for the sustainable future. Today, let's get to know about one of such. Here is the title of my research: Efficacy of nutrient nanoparticle infused slow release fertilizer based on sodium alginate and polyvinyl alcohol for chili plants. The need for food is growing along with the world's population. Overuse of fertilizers increase the environmental, economical, and health issues instead of cope up this food insecurity. This kind of present scenario leads the modern agriculture industry towards creating an eco-friendly and cost-effective, innovative fertilizer. One of such fertilizer called as slow-release fertilizer. What is slow-release fertilizer? In short, it can be defined as fertilizer with a brain and intelligence, cause that apt to slow down the nutrient release to be more in synchrony with plants' need throughout growth season. Here, some of the advanced features of slow-release fertilizer over conventional fertilizer are listed. Basically, the fertilizer productivity is enhanced dramatically by high nutrient use efficiency in the slow-release fertilizer type. This is a key feature that driven the other features also. Customize the nutrient type and amount will be an added advantage in the synthesis of slow-release fertilizer. Nitrogen, phosphate, potassium, magnesium, and zinc are the nutrients that are incorporated in the new formulation of slow-release fertilizer in this research. These are the nutrients basically need for any plant. Among them, magnesium and zinc were synthesized as oxide nanoparticles using sol gel method. Here is the detailed pictorial representation of the method with the required conditions. After the series of process, the obtained nutrient nanoparticles were characterized using XRD, FTIR, and scanning electron microscope. FDR spectra of both nanoparticles shown here confirm the successful synthesis of nanoparticles. The peak named as 7 indicates metal-oxygen chemical bonding. XRD spectra of both nanoparticles exhibit diffraction peaks corresponding HKL values of zinc oxide and magnesium oxide. Average crystalline size of zinc oxide and magnesium oxide calculated as 31.19 nanometer and 17.40 nanometer respectively. SEM image of zinc oxide nanoparticle clearly observed to be spherical in shape. The irregular small clusters of aggregated zinc oxide nanoparticle and magnesium oxide nanoparticles were observed. Homogeneous mixture of both polyvinyl alcohol and sodium alginate polymers were blended with selected nutrients under required conditions. Final polymer blend was added dropwisely into the cross-linking solution in order to prepare the slow-release fertilizer in the form of beads, as shown here. The FDR peaks intensity of slow-release fertilizer bead shows the fine chemical bonding between the corresponding species. Same image of slow-release fertilizer beads depicts a partial fulfillment of required standard. Swelling ability and nitrogen release of slow-release fertilizer bead were measured and received within the required level of rate. It is essential for slow release of nutrient. Chili plant was selected as a model crop and treated under 4 distinguished treatments which are given here. In a nutshell, plant growth, fruit yield, and nutrient uptake of slow-release fertilizer bead treated plants significantly differ from rest of the group. Statistical analysis of the data also confirm that. Finally, I conclude with the research findings that are listed here, which confirms the potency of this advanced and innovative concept in improving the crop yield and quality. These are some ideas for future work that require to implement this core concept of slow-release fertilizer in a large scale into the agri industry. Thank you for listening and spending your valuable time on my presentation.
Okay, thank you very much, Ms. Tonya. The paper is open for discussion. Please, any comments or clarification?
Miss Tanya, have you done a cost benefit analysis of these compounds?
Actually, madam, the cost for these the materials will be like it was higher than the normal conventional fertilizer. But when we thought about the future scenario, like if the post harvest and the post scenario, it will be very cost effective for the farmers and the agriculture system when we consider about the conventional fertilizer.
Cost benefit analysis means higher means you mean the cost is higher or more benefit? Higher cost
Like
for
cost meaning the synthesize the mechanism, the machine, the equipment. Yeah.
For nanofertilizer treatments, cost was higher.
Yeah, like the equipments and the analysis mechanism, machinery, those kind of stuff is higher. But when we talk about the post harvest and the post field treatments like that, it will be very cost effective when consider the conventional and traditional fertilizer.
You have done, Hamad, how long you have done this experiment? How many times that One growth cycle?
Yeah, it's a one growth cycle.
For how many months?
It's about 3 months.
3 months.
Yeah, the cycle, the cycle, the growth cycle is 3 months, but the research period is 6 months.
Have you find that any that extent of growth period when you use this slow releasing fertilizer?
Actually, because of the time constraint, I couldn't do that. But it should be for the future. If the future works can be lead like doing a third, fourth harvesting cycles, that will be more results precise. Will be.
Okay. Any other comment, please? Okay,
Yes,
thank you.
please.
Sorry? Yes.
Yeah, and a small clarification. Yes, Miss Tanya. So you focusing regarding the post-harvest. It will be reduce the post-harvest losses with compared to the other type of fertilizers which we are using for the capsicum and in chili. So can you explain further? Did you analyze or did you check the differences of that post-harvest losses reduction from what you are suggesting?
When we doing the nutrient analysis for the yield, like a fruit, chili fruit, when the treated like slow release fertilizer treated chilies are very nutrient high nutrients than the other treated, and also the environmental loss of the fertilizer also very low. So those kind of economical and agricultural benefits are in the slow release fertilizer.
Okay.
Right. This composition of that different nutrient and that conventional use one and whatever that you mentioned, this slow release one are more or less same, right? Composition, NPK, calcium, magnesium.
Yeah.
Same or different?
Are you asking about the conventional fertilizer and the slow release fertilizer?
Yeah. Now what you apply?
Yeah, it's more of the composition is similar, but the way we adding the fertilizer, it's in the polymer hydrogel slow release fertilizer.
Yeah.
There it's a solid and liquid stage fertilizer.
Right. You mean that amount you applied for different treatment also more or less similar?
No. Yeah,
different.
it's similar. No difference. It's similar. The way we apply it is different.
Yeah. How you can explain this? The protein content is higher, or the nutrient content is higher in slow release in fertilizer.
When after the harvest of the chili fruits, we are doing the proximate analysis for the chilies. Through that, we are getting the ash content and the carbohydrate protein contents. So when we are analyze that, the chilies treated with slow release fertilizer is higher than the other variety.
That is true, but I mean, how you can explain why that what process that why, what are the reasons for increasing this nutrient content or whatever the protein content?
Yeah. So so the slow release fertilizer basically made up with a super absorbent polymer. So polymer can easily absorb hold a large amount of water and the nutrient contained water up to several hundred times than its own weight. So when the fertilizer is applied, the polymer will release the water and the nutrient slowly over time. But in case of the traditional fertilizer, it is contrast. Like it can be released as one time, so all at one time. So that's a difference. Here the slow release capacity helps to prevent the nutrient from washed away by train and leaching into the groundwater.
Have you
yeah.
Determined the nutrient use efficiency of different formulations and that conventional and slow releasing? Whatever do you
Sorry,
apply, how much they use?
Can you ask again that question, sir?
Sorry? Nutrient
I
use
couldn't.
efficiency of different treatments. Have you calculated or analyzed nutrient use efficiency?
No, sir. It not. I didn't do that.
No, you can get a better idea of this. Most probably, so relation of more on the higher nutrient use efficiency. Okay, thank you very much. Right. Okay. Thank you, Tanya. Shall we
Thank you,
move
sir.
to
Thank you
the
so much.
next presentation? Next presentation on effects of variable seed rates and potassium levels on yield components and quality parameters of basmati type number 6 rice in Sri Lanka. Miss HMKGSSK Herath is going to present.
Hello everyone, I am Sanjana Sundari Herath from Faculty of Technology, University of Colombo. Here I mention co-authors as well. Our research topic is effect of variable seed rate and potassium levels on yield component and quality parameters of basmati type number 6 rice in Sri Lanka. This is the content of my presentation. Rice is the staple food for over 21 million people in Sri Lanka. Basmati rice stands out among rice varieties due to its fragrance, long grains and cooking qualities. Potassium fertilization enhances grain yield and quality. A suitable seed rate ensures proper plant establishment, growth and development, influencing grain yield. The interaction between the seed rate and potassium fertilizer significantly impact rice yield and quality. Recent studies examine the effect of potassium fertilizer and seed rates on rice varieties shown in this table. As you can see, no research have done to evaluate the interplay between the potassium fertilizer and seed rate on basmati rice in Sri Lanka. Therefore, this research aimed to address this gap by optimizing these factors for the newly developed basmati type number 6 rice variety from CIC Agribusiness. The overall objective of this research is to identify the suitable seed rate and potassium fertilizer combination on grain yield and quality of basmati type number 6 rice variety. The specific objectives include to identify the most suitable seed rate and MOP fertilizer level and to identify the most effective seed rate and MOP fertilizer combination on grain yield and quality of basmati type number 6 rice variety. Materials and methods. The research project was conducted at the Rice Research and Development Unit of CIC Agribusiness at Palwera during yellow season. Research materials were used as the certified seed paddy of basmati type number 6 variety, fertilizer, herbicides and pesticides. These are the treatments and experimental layout of this study. In here, we were tested 3 different seed rates with 3 different MOP fertilizer levels. This is the field layout of the study. 6 by 3 meter plots were arranged in RCBD with 3 replicates. Methodology. First land preparation was done. Then pre-germinated seed paddy was broadcasted. Pest and disease management and fertilization was done. Harvesting was done at 85% maturity age. Finally, post-harvest operations were done. Data collection, vegetative parameters, yield component, quality parameters such as physical, milling, and cooking quality parameters were recorded. Data analysis, two-way ANOVA was used to see the significant differences among the tested seed rate and potassium fertilizer levels using SAS. Mean separation was carried out to see the significant differences among treatment using Duncan test at 5% significance level. Results and discussion. Tested MOP levels and seed rate do not have an effect on plant height and flag leaf length. 45 gram seed rate has significantly increased the panicle length and number of productive days. When consider the combined effect, 45 gram seed rate with 1.5 of DOMOP treatment significantly achieve maximum panicle length, and same seed rate with no MOP treatment significantly achieve maximum number of productive tillers. 135 gram seed rate and 45 gram seed rate significantly increase the panicles per square meter and spikelets per panicle. Increase in MOP level up to 1.5 of DOA MOP has significantly increased the 1000 grain weight. When consider the combined effect, 45 gram seed rate with no MOP treatment and 135 gram seed rate with 1.5 of DOA MOP treatment significantly increase the spikelets per panicle and 1000 grain weight. Tested MOP levels and seed rate do not have an effect on actual yield. 1.5 of DOA MOP has significantly increased the grain length. 1.5 of DOA MOP has significantly increased the head grain percentage and significantly decreased the broken grain percentage. 1.5 of DOA MOP and 90 gram seed rate significantly increased the before and after cook length. According to the combined effect, 90 gram seed rate with 1.5 of MOP treatment significantly increased the quality parameters. Based on these findings, the study concluded that 90 gram seed rate per plot with 1.5 of DOMOP is the optimal combination to achieve maximum yield and quality for Basmati type number 6 rice variety. Study should be repeated in another Yala and Maha season to ensure that the findings. These are the key references that support for this research. This is the end of my presentation. Thank you for your attention.
Okay, thank you very much, Ms. Hera. Right? Now the presentation is open for discussion. Just clarification, Ms. Hera, this is the factorial design, right?
Yes.
Then when you explain the results, you mention sometimes that combined effect, sometimes individual effects separately. But you didn't mention that time that whether we don't know exactly is there any interaction for some. I feel that the same data set you mentioned sometimes combined effect as well as individual effect, right? Have you find any interaction effect? Combined means interaction effect, right?
Yes, sir.
Factorial. No? So have you find that interaction effect for all the parameters?
No, sir.
What are the parameters you have found interaction effect?
In quality parameters, sir, cooking and physical quality and cooking quality parameters.
Only.
Yes, sir.
Except this potassium and potassium fertilizer, you keep the agriculture department recommendation, right?
Yes, sir.
And plus some potassium. Your conclusion was adding potassium fertilizer, you got more benefit, higher yield, higher quality, like that. Correct?
Sorry, sir.
And that your conclusion mentioned that when you applied more than recommended fertilizer, you get higher yield, higher quality parameters, like that. Is that correct?
Only
this finding, study concludes that using 90 grams seed rate per plot, 1 bushel per acre, with 1.5 of the Department of recommended MOP is optimal combination to achieve maximum yield and quality of basmati. you got the higher yield and higher quality when you applied more potassium fertilizer than recommended. Correct?
Only for quality, sir. Quality parameters.
Then your abstract mentioned, based on these findings, the study concludes that using 90 gram of citrate per plot, 1 bushel per acre with 1.5 of the department of recommended MOP is the optimal combination to achieve maximum yield. Those are not the maximum yield means not the qualitative parameters. Right? Correct? Is that correct or not?
Not,
Not correct?
sir. Maximum.
Please, let me conclude your experimental result. Just to conclude, what is your conclusion?
Sir, yield is same for three different seed rate.
Yeah.
Therefore, I conclude.
Then fertilizer. What is the influence of fertilizer on yield?
Yield is the same, sir, in three different MOP fertilizer levels.
There was the wrong conclusion here in abstract. Abstract, you mentioned higher yield with 1.5 of the department of recommended MOP. Okay, check that.
Yes, Miss Serath, according to that, according to your result, the conclusion is there is no any significant difference with the recommendation which gave by the Department of Agriculture. Is it?
Yes, ma'am.
That means although you increase the MOP rate or although you reduce the seed rate, there is no significant difference.
But can you check the abstract? You check the abstract.
In the abstract, you mentioned that what the professor said in your abstract, you mentioned the half rate of seed.
1.5. 1.5 of the department of
MOP and
recommended
the
is the
calorie
optimal combination
half
to
1
achieve
bushel
maximum
per
yield.
acre. 1 bushel per acre. That means the half.
Additional
Rate,
50%.
yes, seed rate is with the recommendation half seed rate.
Using 90 gram seed rate per plot, 1 bushel per acre with 1.5 of the department of recommended MOP is the optimal combination to achieve maximum yield. That means that you got maximum yield from that treatment.
Yes, that means that it means
Different from what you presented.
Yes.
Yeah. Please check which one is correct. Ms. Ratna. Just to check. Yeah.
Okay, thank you.
Otherwise, that wrong results. You just check your presentation and the abstract. Okay.
Okay.
Thank you.
Thank you.
Professor, shall we go for the next presentation?
I am Hashini Vijayasuriya from Faculty of Graduate Studies, University of Ruhuna. The topic of my study is Agronomic and Economic Assessment of Top Dressing Timings in Gherkin Cultivation. This is the flow of contents. Gherkin is a high-value horticultural crop in the Cucurbitaceae family. As a fast-growing and high-yielding crop, efficient nutrient management is fundamental aspects of optimizing gherkin production. Top-dressing fertilizer application plays a crucial role in supplying essential nutrients during the plant's active growth phases. The application of fertilizers at the correct time is essential to maximize both yield and nutrient use efficiency. Since gherkin has distinct growth stages, it is critical to align fertilization with the crop's peak nutrient uptake periods. The deficiency or excess of any of these nutrients can significantly impact plant health and productivity as well as farmers' revenue. Therefore, this study evaluates the agronomic and economic impacts of varying top dressing schedules in gherkin cultivation, focusing on the optimal timing of the first top dress application. Next, move to the problem statement. Timely fertilizer application is a key to maximizing yield, nutrient use efficiency and profit, while minimizing nutrient loss and input cost. However, the most profitable timing is unclear. Therefore, both agronomic and economic assessment are needed to determine the optimal application time in gherkin cultivation. The main objective was to assess the economic and agronomic impact of different top dressing timings in gherkin cultivation. Next, move to materials and methods. We used the gherkin variety Kirti F1. Standard cultural practices were followed. In harvesting stage, grade 1 gherkin fruits, which are 11 to 14 mm in diameter, were harvested. The treatments were based on the timing of the first top dress application. Subsequent top dressing were applied every 10 days up to 60 days. This is the experimental layout. We collect both agronomic and economic data. Data were statistically analyzed using one-way ANOVA with SAS software. Means were compared using Duncan's multiple range test. Next, move to the result and discussion part. Starting with vine length at 50% flowering stage, T1 and T2 showed significantly highest vine length compared to T3 and T4. Vine length at first harvesting stage, also T1 and T2 showed significantly highest vine length compared to T3 and T4. In terms of leaf area, the 14th leaf showed the significantly highest size in T1, T2 and T3. However, for the 21st leaf, T4 surprisingly had the significantly highest leaf area. In reproductive parameters, T1 and T2 had the highest number of flowers per vine. Also, T1 and T2 had the highest number of fruits per vine. Also, yield per vine is significantly highest in T1 and T2. In economic analysis, we calculated material cost including fertilizer and labor. This is the total production cost for 1 acre. Revenue was calculated based on market price, 240 rupees per kilogram for grade 1 gherkin. When comparing the gross and net benefit, T1 and T2 had the highest gross income within T2 showed the highest net benefit. T4, despite having lowest yield, showed highest cost-benefit ratio due to its minimal input cost. Finally, in the conclusion, we can conclude that starting top dressing at 10 days lead to highest growth and yield, but also highest cost. 15 days give the best balance of profitability and productivity. Delaying top dressing to 20 to 25 days can save cost, but yield is significantly reduced. Therefore, T2 is recommended for both agronomic and economic return. These are the references I used in the study. Thank you for your attention.
Okay, Ms. Vijayasuriya, there's any comments or questions on the presentation? Hello.
Yeah, thank you, Ms. Vijay Surya. I have one concern that what is the farmer's general practice for in relevant to that jerk in top dressing? What is the date they have put that your top dressing?
Normally they use 15 days as first top dress due to recommendation for salad cucumber under protected house conditions.
So it means that throughout your research, you have concluded that the 15 days is the most efficient date applied top dressing, isn't it?
Yes, sir.
Okay, thank you.
You mean same one as existing practice, right? No difference.
Yes, sir. They recommended 15 days for salad cucumber under protected house conditions. Due to that foundation, we find a specific for gherkin cultivation for specific as well as our variety.
You confirm those terms with existing results, right? So why you mentioned in your presentation you use clay soil. Why you use clay soil?
Due to the reducing nematode attacks. When we use a normal soil, it has more cause to nematode attacks. Due to that, we use clay soil.
What is the soil mixture you use? What is mixture you use?
Clay soil, paddy husk, cow dung.
Paddy husk, cow dung. Any issues on drainage?
No.
Have you observed?
Good drainage conditions. We also use the aeration tubes also.
Then in your economic analysis, you mentioned that net benefit and benefit cost ratio. What is the difference? What do you mean by net benefit and the benefit cost ratio?
In net benefit, we calculate total cost, total revenue subtract by total cost. In cost benefit ratio, we use the calculation total revenue divided by the total cost.
You mean better way is net benefit, calculation of net benefit?
Sorry, sir.
What is the best way of that recommend in some of your treatment? For example, T2. You recommend T2, no? What is the what is the cost benefit ratio is different than net benefit.
Cost benefit ratio is best.
Best. You mean that what is the best parameter to recommend the net benefit or benefit cost ratio?
Using only cost benefit ratio, it means T4 have 28.1, but it means not good. It has low input cost as well as low yield. In T2 27, it not mean not good as T4. It means effectiveness of balance between cost and yield also.
Yeah, now you recommend T2, right?
Yes, sir.
T2 recorded the highest net benefit per acre.
Yes, sir.
Right. You mean beta parameter is net benefit ratio.
Yes,
right?
sir.
Correct?
Yes, sir.
Right. That's all. Any other comments, please? So if there is no comment, Thank you very much, Ms. Vijayasuri, for your nice presentation. Shall we move to the next presentation? Increase in nitrogen use efficiency in rice using nitrate and ammonium inhibitors.
Good morning everyone, I am Lakshmi Namanjana. I am going to present my research project on the increasing nitrogen use efficiency in rice using nitrate and ammonia inhibitors. Rice is one of the most important cereal crops in globally and a staple food for over 21 million people in Sri Lanka. Rice requires significant amount of nitrogen nutrient which is crucial for optimal growth and productivity. This is the mechanism of urea absorption of plant. Ammonia and nitrate are the only forms of nitrogen that plant can be absorbed. However, the nitrogen use of efficiency of rice plant is only about 30%. 70% of the applied nitrogen is lost to the environment through processes like denitrification, waterization, and leaching. To address that issues, we can use nitrogen inhibitors. Nitrogen inhibitors slow down the conversion of nitrogen to forms that are susceptible to losses. There are two types of nitrogen inhibitors. The first one is ammonia inhibitors. The second one is nitrate inhibitors. There are the few research that have been done during last 10 years related to the different nitrogen inhibitors. You can see there is real research conducted using nitrogen inhibitors to increase the nitrogen use efficiency in rice, and also no research conducted specifically BG352 rice variety. Therefore, the general objective of this research is to study the efficiency of nitrogen inhibitors to enhance the nitrogen use efficiency of nitrogen fertilizer. This research was conducted at the CIC Agribusiness's Rice Research and Development Unit in Palwehera, Dambulla during yellow season. We used BG352 3.5 months semi-draft new improved rice variety along with necessary fertilizer and pesticide. In here we used 7 treatments. We used a randomized complete block design with 3 replicates. In here we did field preparation and crop establishment. Irrigation was done by surface irrigation method. Then the fertilizer applied according to the Department of Agriculture recommendation. Only nitrogen fertilizer and inhibitors applied in the relevant plot according to the treatment. We collected actual grain yield and yield component, vegetative parameter and estimated parameter. Harvesting was done at 85% maturity stage. Finally, post-harvest operations were done. Collected data were analyzed using 1V ANOVA, mean separation done using Tukey test. In here, you can see vegetative parameter response for different urea levels and nitrogen inhibitor treatment. According to that result, there are no significant difference among treatment except no nitrogen treatment. In here, you can see the effect of different treatments on actual grain yield response of BG352. According to that result, there is no significant difference in actual yield between 100% RDU and 60% RDU with vibasol inhibitor. Nitrogen use efficiency is the very important parameter. In here, consider the effect of different treatment on nitrogen use efficiency. 60% RDU with vibasol inhibitor showed the significantly highest nitrogen use efficiency. This experiment concluded that there was no significant difference in actual yield between DOA recommended of 100% urea and 60% RDU with vibasol inhibitor, and also 60% RDU vibasol showed the significantly highest nitrogen use efficiency. These findings suggest that 60% RDU with vibasol inhibitor is an efficient strategy to enhance nitrogen use efficiency in the rice variety BG352. By adapting this approach, we can reduce urea application by 40% while maintaining optimal yield for BG352 under low country dry zone condition in Sri Lanka. Additionally, among the three nitrogen inhibitors, Vibasol obtained the most significant result in improved nitrogen use efficiency. We want to repeat this study during other Yala and Maha season to validate our finding. This is the reference of my presentation. Thank you very much.
Okay, thank you very much, Ms. Navanjana. The paper is open for discussion. Nice presentation. Just clarification, why you use BG352 variety?
BG352 variety is the most used rice variety in Sri Lanka.
That's why you use, right?
Yes, sir.
I have no much comment on. Any comments or clarification? So we can save more nitrogen, 40% urea, by using urea inhibitor, nitrogen inhibitors, right?
Nitrogen inhibitors slow down the conversion of ammonia and ammonia alteration.
Yeah. Okay, thank you very much. Nice presentation. So,
okay.
Shall we move to the last presentation, I think?
Thank you, sir.
Thank you. Evaluating the effect of different fertilizer formulations on pod filling in the groundnut variety Lanka Jambu by KHMSS Vijay Suru.
Hello, good afternoon everyone. I am Sasit Sandarwan. Today I will present our research on evaluating the effect of different fertilizer formulation on pod filling in the groundnut variety Lanka Jumbo. This is the presentation outline. Groundnut is a globally important oilseed and legume crop. In Sri Lanka, the Lanka Jumbo variety NKG2 is popular among farmers and exporters. It was released in 2015 by the Grain, Legume and Oil Crops Research and Development Center, Agunagola in Sri Lanka. Previously, Sri Lanka imported most processed peanut from China. With Lanka Jumbo, local production now meets part of this demand. Pod filling in groundnut is influenced by various factors including nutrient management. Among them, fertilizer application plays a key role in achieving optimal yield. Despite the existing fertilizer recommendation by the Department of Agriculture, pod filling in Lanka Jumbo remains suboptimal. Therefore, the objectives of this research were to study the effect of different fertilizer mixtures on pod filling of groundnut variety Lanka Jumbo, to identify the best nutrient mixture for growth and development. The material and methods. This study was conducted in Ridimale at the DS Division, Badula District, Sri Lanka. A randomized complete block design was used with 7 treatments and 3 replications. Treatment allocation. This table outlines the 7 fertilizer treatments including: first one is the current Department of Agriculture recommendation. This is a control one. Modified version of it, and selected commercially available complex fertilizer. Data collection. Data collected included plant height, number of branches and yield parameters, as well as growth measurements were taken at 25 and 60 days after planting. Data analysis. Data were analyzed using ANOVA in Minitab with Duncan's multiple range test for mean separation. Then I will move to the result and discussion part. Effect of different fertilizer mixtures on plant growth. This graph shows the effect of different fertilizer mixtures on plant growth. Fertilizer mixtures significantly influence growth at 25 days after planting. However, there was no significant effect on plant height or branch number at 60 days. The effect of different fertilizer mixtures on pod weight. For pod weight, both wet and dry pod weight per plant were significantly affected by the fertilizer mixture. Treatment 4. The Department of Agriculture recommendation combined with 250 kg per hectare of gypsum resulted in the high pod weight per plant. In this slide, you can see the effect of fertilizer mixtures on the number of pods per plant and pod filling percentage. Treatment 4 produced the high number of pod per plant and the high pod filling percentage, recording 96.2%. Similarly, treatment 4 recorded the high 100 seed weight, while treatment 7 performed lowest. This may be because gypsum supplies calcium, and studies show calcium, potassium, and boron improve pod filling and yield. Finally, we can conclude that this result as the vegetative growth of groundnut variety Lanka Jumbo was not significantly affected by the different fertilizer mixtures. But treatment 4, Department of Agriculture recommended fertilizer mixture and 250 kg gypsum per hectare resulted in the high performance across all yield attributes. Applying complex fertilizer alone was insufficient for an effective pod filling in groundnut variety Lanka Jumbo. Those are the references. Presentation is over. Thank you.
Right. Thank you very much. Nice presentation. Mr. Yesurya. Mr. Yesurya, now the paper is open for the discussion. Any comments or clarification, please?
Yeah, yes, thank you very much for the nice presentation, Mr. Vijay Surya. So I have a very small clarification regarding your presentation. So have you identified any effect of your treatment for the flowering characters? So then because of you are not mentioned anything regarding those. So because of some treatments are included here with the high percentage of MOP likewise. So then
Thank you. Normally, development is a safe treatment.
Yes, sir, you're not clear.
Fertilizer flowering state is normally different, not any difference in the research.
You mean different fertilizer formulations are not significantly different, right?
Yes.
Is that the question you asked? Yes, superintendents?
the number of days for flowering is also the same. No any differences with the
Yes.
with the treatment.
Yes.
Okay, right. Okay, thank you.
Just a small comments. What what is the difference between fresh weight and dry weight of food?
Normally, Department of Recommendation, urea, TSC, MOPI, and adding the gypsum.
No. What is the difference between fresh weight and dry weight of pods? You mentioned in your that measuring parameters, fresh weight of groundnut pods and dry weight of groundnut pods.
Yes.
That how you? You mentioned in your that yield parameters, you measured fresh weight and dry weight of groundnut pods. My question was, what are the differences? Any differences between fresh weight and dry weight of groundnut pods?
Harvesting in the groundnut, pressure is very high. Dry weight after
what is the moisture percentage at harvesting?
Dry weight is by moisture 80%. Normally, harvesting in the harvest moisture normally increase of the 10%.
What is the percentage of moisture at harvesting?
Yes, sir.
What is the percentage of moisture at harvesting?
Normally 10 to 14%.
10 to
14%.
10 to 14.
Yes. After the time,
it reduced to 8. After drying, it reduced to 8%.
Reduced to 8. Normally, I check the 8% in the dry weight.
Right. So the other question is, why you use gypsum as calcium source? Stand reason.
Normally, groundnut in the Lanka Jumbo NKG2, is very low variety. Groundnut pot in the field easily available in the calcium and magnesium sulfate for the groundnut pot in the this NKG2 variety. This is the reason.
Easily available means easily available to plants.
Sorry, sir.
What do you mean by easily available calcium source?
Easily available means calcium
to the plant.
In the no market.
No market. No other source. Other sources like that lime, dolomite, we can use now.
Calcium gypsum was included calcium 23% and sulfur included 70% in gypsum fertilizer.
Yeah, that is true. But in your that present, you mentioned that you use gypsum as calcium source. That is why I asked. Right. Any other comments or questions? So if there is no question, thank you very much, Mr. Yesuri, for your presentation. Now I'm that all the presentations are over. Can I wind up the presentation? Just to I'm that few things. I think most of the presenters are final year students from different universities. And most of the experiments generally okay, good. Some are small issues. Otherwise, I'm that comparatively good. There are very good presentation, practically important, timely needed research problems. Also, there are some of the research. But in the final year students, what we expect, not much that like that senior scientist who presented in them or who did the experiment on different problems. But I think they achieved the objectives of the different universities for final year students to identify the research problems or the how to design and the use the design for experiment and then execute the experiment in field and pots, and how to get the parameters, then the statistical analysis and how to present here again and conclude the result like that. I hope almost all the students, they achieve the different universities objective. That is what we are looking for from the students. So I just take this opportunity to thanks for all the presenters and even the supervisors of the presenters and all the audience and Ms. Darshika, so baby Miss, then Rohanadira, sir, thank you very much for thanks.
Okay, thank you, sir. With that, we come to the end of the technical session 1 on crop science and emerging technologies for sustainable agriculture. First, I would like to extend my heartfelt thank to our session chair, Professor S. Subasinghe. Sir, your presence added immense value to the session. Also, I want to express my gratitude to our panel members, Mrs. S.L. Navaratna and Mr. H. Rohanadira, for sharing their valuable thoughts and time with us. Lastly, I would like to thank all the presenters for sharing your findings and contributing to a fruitful discussion. Please join us with the session 2 of the Crop Science and Emerging Technologies for Sustainable Agriculture in a short while. Until then, thank you.
Good afternoon. Welcome you all for the second technical session in our first International Symposium on Agro-Technology and Rural Sciences. The technical session is titled Crop Science and Emerging Technologies for Sustainable Agriculture Session 2. I have honored to invite Dr. N.P. Vidhanapathirana, Senior Lecturer, Head of the Agro-Technology Department of University of Colombo, Institute for Agrotechnology and Rural Sciences, to chair this session. Then I would like to invite panel members, Ms. SAP Nelka, Lecturer, Department of Agrotechnology, UCIARS, and Mr. AJ MCM Sirivardhana, Lecturer, Department of Agrotechnology, UCIARS. Dear Madam, Dr. N.P. Vidhanapathirana, I warmly welcome you to chair this session and proceed. Thank you. Over to you, Madam.
Thank you, and welcome all the presenters for Crop Science and Emerging Technologies for Sustainable Agriculture, session number 2. And we are going to start the session number 2 of Crop Science and Emerging Technologies for Sustainable Agriculture session. And there are several papers we have to discuss and that present. Presenter. That's effect of different rate of ammonium sulfate. The presenter is Miss Arya Soma. Miss Arya Soma, we would like to invite you to present your research findings.
Hello everyone, I am Kalinda Kushan Hinkenda. Here present research finding on selection of suitable rootstock for cucumber cultivation.
Please stay with us a little few minutes. There was a technical issue.
Good morning, everyone. Good morning, everyone. I am Ravihari Arisoma, and I am presenting our research presentation on the topic of effect of ammonium sulfate application on chemical and physical properties in cinnamon growing soils. And here is the flow of my presentation.
To begin with a brief introduction, the cinnamon cultivation requires intensive nutrient management due to the continuous removal of biomass during harvesting. Both organic and chemical fertilizers play a crucial role in maintaining soil fertility. The recommended fertilizer ratio for cinnamon is 2:1:1 NPK, with nitrogen being a key nutrient for shoot regeneration. Among nitrogen fertilizers, the ammonium sulfate is widely used due to its dual role in providing nitrogen and sulfur. Today, cinnamon growers apply ammonium sulfate primarily where they need supplemented nitrogen to meet nutritional requirements, but its long-term effects on soil is yet underexplored. As an example, its long-term application can lead to soil acidification, affecting nutritional availability. So it is not recommended for the wet zone areas in Sri Lanka due to its acidifying properties. So this study investigates the impact of ammonium sulfate on soil chemical and physical properties in cinnamon cultivation, aiming to optimize fertilizer management for sustainable production. The study was conducted at the National Cinnamon Research and Training Center, Thiagoda, located at the Madhya district of Southern Province, and there were 6 treatments, as you can see, varying amounts of ammonium sulfate and urea levels. And as you can see here is the experimental layout with 4 applicates arranged perpendicular to the gradient, and when it comes to the soil sampling, it was conducted in 2 phases to assess both long-term and short-term effects of ammonium sulfate application. And the first sampling was performed 5 months after application to evaluate the long-term effects, and the second was carried out 6 weeks post-application to assess the short-term effects. So the samples were collected from predetermined sampling points within each treatment, targeting both vertical and horizontal distances within the cinnamon root zone, and the soil was sampled at 3 lateral distances from the plant base, 15, 30 and 45 centimeters, and the tree soil depths 10, 20 and 30 centimeters, and the 2 replicates were taken at each depth and distance, yielding 9 composite samples per treatment, as you can see in the figure 2. So that a total of 216 soil samples were collected across all treatment. The collected soil samples were tested for their physical and chemical properties, and the laboratory analyses were conducted to evaluate both chemical and physical properties of soil samples. The chemical analysis include measurement of soil pH, electrical conductivity and available sulfur, and the physical analysis include determination of soil bulk density and moisture content. When it comes to result and discussion, the application of ammonium sulfate significantly affected soil pH in both long-term and short-term. In short-term, all ammonium sulfate applied treatments showed a significant decline in pH compared to the control. These findings confirm the acidifying nature of ammonium sulfate, especially noticeable within 6 weeks of application. When it comes to soil EC, it was significantly influenced by the treatments in both long-term and short-term, and the long-term data revealed a gradual increase in soil EC with higher ammonium sulfate rates, where control shows a lowest reading. The rise in electrical conductivity correlates with the accumulation of soluble salts due to the prolonged ammonium sulfate application, particularly at higher doses. When it comes to available sulfur, ammonium sulfate application had a significant impact on available sulfur content of the soil. In short term, T5 and T6 both recording higher levels, and these results highlight that direct contribution of ammonium sulfate to increasing the soil sulfur content, that confirming its effectiveness as a sulfur source. In contrast, physical properties such as bulk density and soil moisture content were not significantly affected by the continuous ammonium sulfate application in long term. In conclusion, The continuous application of ammonium sulfate at varying rates significantly affected key chemical properties of soil including pH, electrical conductivity and available salt content, and this application of different urea into ammonia sulfate ratios led to a noticeable pH decline within 6 weeks post-application, and this highlight the necessity of applying lime to maintain the soil pH within the recommended range of 5.5 to 6.5 for cinnamon cultivation. In contrast, the soil physical properties, the bulk density and the moisture content remained unaffected by the long-term application of ammonium sulfate, that indicating the structural soil stability was not compromised under the treatment conditions. So here is
I would like to invite panel members to ask questions regarding the research.
Okay, thank you, Ms. Arya Soma. One of your objective was to find out the long-term and short-term effect of ammonium sulfate to the cinnamon soil. So that actually as a summary, what is the significant effect of ammonium sulfate to the cinnamon growth soil as you identified with the research findings?
Yes, madam. Actually, in short term, that means 6 weeks after 6 weeks after application, there's a rapid changes in pH, EC, and the sulfur content of the soil, cinnamon soil, growing soils after application of ammonium sulfate. So these changes actually can be due to the quick solubility and the reactivity of ammonium sulfate, and especially the pH decline very sharply in high ammonium sulfate treatments that may be due to the ammonium nitrification by the soil bacteria like Nitrobacter. So this process release H ions and it increase the soil acidity. But in long term, that means 5 months after ammonium sulfate application, the pH continued to decrease gradually in plot receiving high doses of ammonium sulfate. But the EC and the available sulfur remained high, particularly in treatments with 100% ammonia and with no urea treatments. So this may be due to the accumulation and the slower leaching of the sulfur over time in regular soil, especially the cinnamon growing soil. So these are the chemical changes. When it comes to physical changes, during the long-term process, there is no change in physical properties with the ammonium sulfate application. So that is due to the chemical, ammonium sulfate is a chemical fertilizer that mainly altering the soil chemistry. So that may be the reason there is no change in the soil structure and the water retention over the period. But if we use ammonium sulfate as a part of broader soil amendment strategy, for example, with organic matter, compost, manure-like things, with regular soil amendments, so then we can observe an effect on soil bulk density and moisture content. So this is the summary of applying the effect of applying ammonium sulfate to the cinnamon growing soil in long term and short term with the chemical properties and the physical properties.
Okay, good explanation. Thank you.
Thank you very much, ma'am.
Thank you, Ms. Arya Soma. Actually, that you are in your research, you are analyzing physical and chemical properties with the application of ammonium sulfate. In your results, it has observed that there is no significant changes in physical properties with the application of ammonium sulfate.
Yeah, ma'am.
What could be the reason for that?
Yeah, madam, actually, the ammonium sulfate is a chemical fertilizer. Actually, it alters the soil chemistry mainly, so it can be the reason that there is no change in the soil structure and the water retention ability to increase or lower the moisture content. But if we use the ammonium sulfate as a soil amendment with a broader soil amendment like compost and manure, as I explained earlier, as a regular soil amendment, then we can observe effect on soil bulk density and moisture content over the time.
Okay. Yes. Thank you very much for your explanations and the presentation. And next, we will move to the next presenter. That's topic is assessing the availability and invasiveness of ornamental plants in Sri Lanka using a quantitative risk assessment framework. The presenter is Mr. M.W.J. Pires. I would like to invite Mr. M.W.J. Pires to present his valuable findings with us.
Hello everyone, I am Vagisha Pires and I am honored to present our research on ornamental plants in Sri Lanka. Together with my supervisor and colleagues, we explored the availability of ornamental plants and their potential threat as invasive species through a structured quantitative assessment framework. Sri Lanka, as a recognized biodiversity hotspot, is home to a wealth of endemic species. However, the increasing popularity of ornamental plants has brought with it a surge in non-native plant imports. While these plants serve aesthetic and commercial purposes, some are showing invasive tendencies that can threaten native ecosystems. Hence, a thorough ecological assessment is crucial to inform regulatory frameworks and support conservation efforts. Our study centers around this key question: how available are exotic ornamental plants in Sri Lanka and what is their potential for invasiveness? We aim to explore this through a structured and data-driven approach. To answer our research question, we set the following objectives. First, to identify native and exotic ornamental plants currently in the Sri Lankan market. Second, to assess their potential invasiveness. And third, to explore the legal framework surrounding their import and sale. Our fieldwork was conducted in 6 districts: Anuradhapura, Badulla, Gampaha, Kandy, Kurunegala and Puttalam. These districts represent various bioclimatic zones of Sri Lanka. We selected 2 nurseries from each district, giving us a total of 12 for the study. This map shows the selected districts across the country, covering diverse climates, ensured that our findings were representative of the broader national context. We developed a framework based on the Australian Weed Risk Assessment tool to evaluate plant invasiveness. We interviewed nursery owners using a structured questionnaire and observed market trends. In parallel, we reviewed Sri Lanka's legal documents governing plant trade. The framework includes 10 evaluation criteria ranging from seed production and dispersal methods to global invasiveness and local impact. Each factor was scored from 1 to 5, with 5 indicating a high risk of invasiveness. For example, a plant that produces over 10,000 seeds annually would receive the highest score in seed production. To reflect the varying importance of each criterion, we applied weighting factors. This ensures that each element's influence on invasiveness is appropriately accounted for in the final score. We calculated the cumulative invasive potential index, or CIPI, by multiplying its score by its weight and summing the results. Importantly, older species with long established non-invasive histories were excluded, and focus was placed on newer exotic introductions. As you can see here, Our surveys recorded 255 plant species. Of these, only 24 were native. Exotic species clearly dominate the nursery market. Surveys show that maintaining exotic plants is generally more challenging than caring for native species. Nevertheless, due to consumer demand, nursery owners tend to prioritize selling exotic plants over native ones. Both native and exotic plant sourcing practices varied widely according to the types of plants. This figure shows the countries from which exotic plants are imported. We identified 255 plant species, but only 46 were selected for invasive potential analysis. As I mentioned earlier, we excluded 185 plant species from this study due to their long history without being invasive and some due to the lack of data to continue the analysis. These are the cumulative invasive potential index values with each criterion for 15 most invasive plants that we found. Using our framework, we identified 11 species with a CIPI above 4.5. Notable examples include Lantana camara and Miconia calvescens, both already classified as invasive in multiple regions. Others like Prunus lorocerasus and Euphorbia tirucali also showed high invasive potential. Most nursery owners were partially aware of regulations. Only one nursery was fully registered under the Seed Act. Although Sri Lanka has a strong legal foundation such as the Plant Protection Act number 35 of 1999, enforcement is currently weak. 2 nurseries were found to be selling invasive plants. Our results show that most ornamental plants in nurseries are exotic, raising concerns about long-term ecological impacts. Strengthening enforcement and promoting native plants is essential. To conclude, the dominance of exotic plants in nurseries combined with weak regulation poses a serious threat. 11 species had high invasiveness scores, signaling future risks. We recommend urgent steps to balance biodiversity protection with the growing ornamental plant industry. We propose a series of actionable steps: establish a centralized invasive species database, strengthen import checks, and increase penalties for violations. Promote awareness, support native plant cultivation, and enhance cooperation between stakeholders. Community-based tools like apps and reporting systems can support ongoing monitoring. These are some of the key references that informed our research and framework we developed. So that's the end of my presentation, and thank you very much for your time and attention.
Okay, thank you, Mr. Pires, for presenting your valuable findings with us. And actually, it's a very timely, relevant topic because invasive management is a global environment concern. So now the presentation is open for the discussion. I would like to invite our panel member, Mr. Sirivardhana, joining online. Do you have
Yes,
any
madam.
questions?
Yes, madam. Thank you, madam, and thank you, Mr. Vagesha, for your interesting and, as madam mentioned, timely, important research. So I have a small question. So you mentioned that Lantana camara identified as invasive, highly invasive species already established in Sri Lanka. So yes, we know that. And I wanted to know whether these species they are selling in these plant nurseries are identically similar to these established wild accessions, or are these plant developed, they are developed cultivars introduced by other countries. Did you got my point?
Sorry, sir. I actually.
Yes. I wanted to ask, clarify. You mentioned that Lantana camara identified as highly invasive species. So we know that plant invasive grow everywhere in the country. So I wanted to know whether these species they already selling in nurseries are identically similar to wild species, or they are are those things, they are developed cultivars. Genetically modified cultivars or identically wild cultivars. Is it clear?
Yes, sir. Actually, sir, what we found was the wild variety of the plant, and it was found in Anuradhapura. They were selling that one, and then we didn't go beyond that to identify which if it is a genetically modified one or not. So we believe that it is the wild one. Based on the results that we found through observations.
All right, okay, because I also seen some Lantana camara species. They modified the species selling by the nurseries. So I wanted to clarify whether these are genetically modified things or identically wild cultivars. Thank you, Mr. Vagesha. I suggest you to continue your research up to the genetic level. Thank you.
Thank you, sir.
Thank you, Mr. Srivardhana. And I have very small question that in your research, you mentioned that out of collected species, 185 species were excluded without further studies, okay? And that I would like to know why that, what is the reason for that?
Yes, madam. Mainly because we didn't didn't include them because they were introduced in very long ago.
Maybe you are not having any data, any
And
details
they have
about
not
those
shown
species?
invasiveness. Yes, and also, excuse me. Sorry, madam.
You couldn't find any details about those species?
Yes, some of the we use this model to get our values. So that model requires a lot of data, which was really hard to find, and we had to verify those data, and some of the data were not available through any kind of research previously done. So that is why we had to exclude them. And we think that some of these excluded ones can also be invasive, but still we cannot disclose them as invasive plants until we have the evidence.
Yeah, yes. And thank you very much for your explanations and the study. And thank you. And next, we will move to the next presentation. And that is relative chlorophyll content values for the characterization of Cucumis melo and Cucumis sativus genotypes in Sri Lanka. The presenter is Miss D.W.N. Madhushika. I would like to invite Miss Madhushika to present your valuable research findings with us. Okay, sorry, the presenter is Mr. P.R.S. Pathirana, and I would like to invite Mr. P.R.S. Pathirana to present his valuable findings with us.
Hello everyone, I am P.R.S. Pathirana, the presenting author of this abstract. Other authors of this abstract are Dr. E.Y. Fernando, Dr. D.M.D. Disanayaka, Dr. K.R. Bandara, and Professor A.M.J.B. Adhikari. We are from Rajarata University of Sri Lanka. Our abstract topic goes as relative chlorophyll content, SPAD, values for the characterization of Cucumis meloel and Cucumis sativus genotypes in Sri Lanka. The introduction of the abstract. Cucumis meloel von kakiri is a significant vegetable species in Sri Lanka. In Sri Lanka, kakiri comes under the broader species of sea mallow, which encompasses a wide array of subspecies, cultivars, ecotypes. The complexity and the diversity within sea mallow make it crucial to understand the species delimitation of kakiri within the Sri Lankan context, which compared to sea sativus or cucumber cultivars for breeding and conservation purposes. Here we report the applicability of SPAD for characterization genotypes. Materials and methods. 8 sea mallow genotypes and 12 sea sativus genotypes were planted for the field assessment. SPAD measurements. The data collection for chlorophyll fluorescence based photosynthetic traits started 4 weeks after planting and measured 2.5 months using the handheld multispec V2 series device. One plant per replicate was sampled and determined the chlorophyll fluorescence and photosynthetic traits from 1 middle and 1 upper canopy leaf. And the photosynthetic measurements were uploaded to Photosync platform that is available in following link of the Google. The statistical analysis of SPAD data. The daily sets of SPAD data that the total value was 17 were subjected to correlation that was given in figure 1b in the next slide and principal component analysis PCA in Minitab 16. The biplot of two major principal components was used to depict diversity based on SPAD. Figure 1c in the next slide. The PC loading status of the PC1 was evaluated and two major determinants, which days of SPAD data, were used to depict given in figure 1d in the next slide and validate the diversity structure created by two major principal components. Here you can see the related diagrams. Results, discussion and conclusion. The daily set of SPAD measurements were positively correlated starting from the second day of SPAD measurements. In PCA, PC1 and PC2 explained 35.1% and 13.8% of the total variance generated by the entire array of daily SPAD data. PC biplot demonstrated that the C. mellow and C. sativus genotypes got clustered separately except for Fiesti, Weldy, Sassy and Thiembra white of C. sativus. PC1 was mainly affected by SPAD readings of this particular date and PC2 was affected by that particular date minus 0.44 value. When we drew a scatter plot between the SPAD data of that particular date, and 17th of 7 in 2022, species-based clustering except for yellow sea small with C. sativus and PST with C. mellow. The present study demonstrated that high throughput SPAD measurements can be used to characterize the Cucumis mellow and Cucumis sativus genotypes, reflecting their species delimits. My reference have been given in this slide. The complexity and the diversity within C.
Thank you, Mr. Patirana, for your nice presentation and valuable findings. And it's a good study. And you clearly state that taxonomic and breeding importance of Cucumis mellow in Sri Lanka and the importance of doing the research. And therefore, it's a feel that it's a valuable research. And I would like to invite panel members to ask questions.
Okay, thank you, Mr. Patirana. I have to clarify a small thing that are there any genetic relationship in between these clusters?
Yeah. In another chapter, we have studied about the genetic or morphogenetic relationship between these species, Cucumis mellow and Cucumis sativus, and also including the species delimitations unclear Thieberra-like variety. So that is actually a separate thing, and we could find, and we have done the sequencing part, and we have targeted 2 molecular markers. By considering those phylogenetic trees and all, we could find clear characterization among molecular level characterization among these varieties, species. Yes.
Okay, then how we can apply these findings to the plant breeding program practically?
Yeah. You mean in molecular level and also photosynthetic level, all these things,
Yes.
right? Yeah. So we have considered a few different indicators like photosynthetic things, spare these things, and also molecular things. Considering all these things, when you think about the Cucumis sativus genotypes, germplasm, there are significant opportunities for enhancing crops by doing breedings in between them. As an example, cultivar Decia pipina like species are closely aligned. Their molecular level, their DNA sequences were also closely level. So we can think about by considering their resistance, disease resistance, pest resistance, and all the other characteristics, we can select best plants for breeding programs, obviously, yes. And other than that, gherkin species, we included 6 gherkin species to study, and as commercially valuable vegetable plant, the gherkin varieties can be also utilized to breed with Decia pipina varieties, and we can enhance these things considering different factors like high yield and all these things. Yes, there is opportunity to do that. And
Okay.
including when you think about the Cucumis mellow germplasm, cactus species like in Sri Lanka, we could have done germplasm collection, island-wide germplasm collection in the baseline first step of the research, and we could find there were actually 4 different cultivars, farm varieties in Sri Lanka. We can also utilize them. They were found in different areas of the country. Some were from Mena, some from Utlan. So we can utilize those distal varieties to cross and make new breeds. By considering salinity and soil levels and all these things. in the Samaran like areas, Verigatha like areas, scenic agar species are available. But in case of Nura, Candy, Kurunagal like areas, we can't find such varieties. Some people know aware about these things. So they know only cooking category, cooking variety. So we can develop new varieties by considering all these by germplasms. We can develop new varieties to cultivate in distal areas like Kurunagala, Kalambu, right? We have done that. We have identified that relationship. Yes.
Okay, thank you for your detailed clarification. Thank you.
Yes, thank you, Mr. Patirana. And I have a very small question. Did you observe any environmental or growth stage effect? That means flowering or fruiting stages. Any difference observation in SPAT reading?
Sorry, last
Did you observe any environmental or growth stage differences when you are reading SPAT chlorophyll content?
That's in the very first weeks, since the chlorophyll this thing has not well developed, there are no significant difference between these varieties. Spat values were not significantly differed, but when they are flowering and mature stages passed, as an example, when the flowering started, they were significantly differed. And before that, when after second or third week, they started to given significant characterization. How can we say? The span values were significantly different among these varieties. Especially, they were divided into ISATRS and Melo clearly. And in the later stage, at the growth, the difference was again minimized. The reason might be due to the degradation of the chlorophyll molecules and all these environmental factors. Yes, again, this came into the peak at the flowering and fruiting stage. Yes.
Yeah. Yes. Thank you for the explanation, and thank you, Mr. Patirana, sharing with your valuable finding with us. And thank you. And we will move to the next presentation and next research. And that is impact of different concentration on gibberellic acid on the growth and yield of rice under low country dry zone in Sri Lanka. The presenter is Miss D.W.N. Madhushika. I would like to invite Miss Madhushika to present her valuable findings with us.
I am D.W.N. Madhushika. Today I will present my research on impact of different concentration of gibberellic acid on the growth and yield of rice under low country dry zone in Sri Lanka. This is the content of presentation. Introduction. Oryza sativa belongs to Fabaceae family. Rice is the staple food of half of world population and a major crop in Sri Lanka. Gibberellic acid is the most important growth regulator. This regulator involved in internodal cell division and plant height. Problem justification. Hormones such as gibberellic acid are used by farmers to enhance the growth. Limited scientific validation is available in Sri Lanka. Therefore, conduct that research. The research major objective was to study the effect of different concentration of gibberellic acid on the growth and yield of Oryza sativa. Its specific objective is to identify the best concentration of gibberellic acid for rice cultivation. Experimental design was randomized complete block design with 4 replicate and 6 treatment. This study conducted at Rice Research Station Ambalanthota using 80 Tulsi Hattabeka rice variety. Next, move to the methodology. Nursery preparation. First, nursery was prepared. Then, after 14 days, pot were filled and transplanted. Fertilizer and irrigation were applied According to the department recommendation, application of gibberellic acid. Gibberellic acid treatment were applied 4 weeks after the date of planted. Before applying, concentration level was calculated. 15 milliliter solution was applied per one plant. Data collection. Data collection was done both growth parameters and yield parameters. This is the collecting parameters. Data analyze. Tested one-way ANOVA using meter statistical software. Mean separation done by LSD at 5% significant level. Result and discussion. Application of gibberellic acid significantly depends on both growth parameters and yield parameters. According to the growth parameters, treatment 5, treatment 6, it means 100 ppm and 125 ppm significantly difference plant height and leaf length. Treatment 4 and treatment 4, treatment 5 and treatment 6 significantly increasing leaf length. Move to the yield parameters. Under the yield parameters, 75 ppm, it means treatment 4 significantly increase all the yield parameters. Conclusion. According to the result, application of gibberellic acid 100 ppm and 125 ppm will significantly increase the plant height and leaf area of the rice plant. Application of gibberellic acid 75 ppm, 100 ppm and 125 ppm significantly increase leaf length of the rice plant. Gibberellic acid 75 ppm has been shown the promising result for yield all the yield parameters. Therefore, application of gibberellic acid 75 ppm can be effectively enhance the yield of rice. This is the references of research. Thank you.
Thank you, Ms. Madhushika, for your nice presentation and sharing valuable findings with us. Actually, that a good study, and GA3 is widely recognized growth regulator, and the application of rice is also promising. And I would like to invite our panel members to share and comment on the presentation.
Okay, Ms. Madhushika, generally, what is the effect of GA3 for plant growth and development? Generally, in general, what is the GA3 effect of GA3 for plant growth and development? How it's affect for the development of plant growth parameters or the yield parameters generally? What happened with the GA3?
High concentration level and generally increase the plant height and leaf area like parameters.
Okay, then in your study, you have selected a few treatments and some concentrations. So what was the base? How you determine those concentrations and the treatments?
According to the literature.
Okay. Then there were some reports or the public data for the GA3 application for the rice.
Yes.
Okay, then according to your study, what was the main effect or the main observation you have done with your study?
75 ppm level increase the yield parameters, negatively affect
We couldn't hear you.
according to the our finding. 75 ppm level enhance the yield parameters.
Okay. Thank you very much.
Okay, Ms. Madhushika, I have a very small question, and that you recommended that 75 ppm is the giving high yield, and it's recommended as a good result in treatment. Okay, and did you evaluate the cost benefit analysis of this research? That means application of GA3 in 75 ppm, and based on the cost analysis, cost benefit analysis, is it viable?
Not analysis.
Not done. Yeah, better that if you can calculate the cost benefit analysis, whether we can apply that in the field level, okay? And whether the farmers can afford that cost, and whether they are gaining something by application of this one, okay? Comparing yield and the cost. Okay, thank you. And we will move to the next presentation and the next research, that is diversity and pest range of fruit flies in 4 selected commercial fruits in Sri Lanka. The presenter is Mr. T.M. Kavinda. I would like to invite Mr. T.M. Kavinda to present his valuable findings with us.
As an agricultural country, Sri Lankan economy is filled with fruit industry also. But there's a topic less studied in our country, but having a high impact on fruit cultivations, the fruit flies. So I'm going to fill the gap on diversity and pest range of these fruit flies in 4 selected commercial fruits in Sri Lanka. I'm T.M. Kavinda. Decorative fruit flies belong to order Diptera. They are considered as one of the major pests throughout the world, and tropical climate in our country has increased the fruit fly diversity also. And fruit cultivation forms a high revenue for Sri Lankan economy. So banana, mango, papaya and guava are the most cultivated fruits in Sri Lanka and also in the Matra. Fruit industry is an important sector in global and local agriculture. One major impact on fruit industry is fruit flies. So they reduce yield and the quality of the harvest. Also, past statistics on fruit fly pests in Sri Lanka are also scarce. So this study aim to identify fruit fly species diversity and pest ranges of those. So I selected 2 objectives: to study the diversity of fruit flies and also to study the pest range of these identified fruit flies. So I conduct this study on from August to November of 2024 inside Midigam Fruit Farm located in Midigam city area. I selected 4 fruit types as mango, guava, papaya and banana. So I selected 2 subsampling sites for each fruit in this study. Having 10 square meters per one study area. So this is the map of the Midigam Fruit Farm, and as you can see, there are 2 subsampling sites for each fruit type. I was selected. Total of 10 sampling rounds were conducted in my study, 1 round per week. So methyl eugenol fruit fly traps was used to capture these fruit flies. So capture was collected after 1 week and counted. 10 drops of methyl eugenol was added at each sampling round. So I observed this capture using simple dissection microscope and identified up to species level using pictorial guides. Then I calculated the abundance, and statistical analysis was done when it is necessary to compare these fruit fly species. So let's move on to is the result. So as the first objective, identification of fruit fly species was done. So I identified 9 fruit fly species belonging to 2 genera. So they were genus Bactrocera and genus Vigoricus. So genus Bactrocera was the most abundant fruit fly genus in this study site, and also it contained 6 species. Genus Vigoricus contained only 3 species. These are the recorded fruit flies in my study, and as you can see, Bacteria genus was the most abundant fruit fly species in each fruit type also. From this recorded fruit fly species, Bacteria dorsalis was the most abundant fruit fly species in all fruit types. Second most abundant fruit fly species was the Bacteria canadensis, also which is endemic to Sri Lanka. In mango, most abundant fruit fly species was the Bacteria canadensis, but in guava, papaya and banana, Bacteria dorsalis was the most abundant fruit fly species. As the second objective, I identified the best range of these recorded fruit fly species. So as you can see, Bacteria dorsalis, Bacteria canadensis, Bacteria correcta, Zygoticus cucurbitae and Zygoticus tau was recorded in all 4 fruit types. But so they have high best range compared to other fruit fly species. Considering all things, genus Bacteria was the most associated with fruits than genus Zygoticus. And also, 3 of these recorded fruit fly species are major pests in Sri Lanka, which are Bacteria dorsalis, Bacteria canadensis, and Zygoticus cucurbitae. And also, these 3 were the most abundant fruit flies in mango, guava, papaya, and banana. Fruit flies have adapted to various fruit types with high pest range. Normally, mango attract much amount of fruit flies. As my study period is not in line with the fruiting season of mango, Bacteria dorsalis abundance was lower, but they survived using other fruits like guava, papaya, and banana as their alternative hosts. Finally, we can say genus Bacteria have larger pest range, so they are polyphagous, and dominant pest on mango was candy fruit fly, which is Bacteria canadensis, and also the dominant pest on guava, papaya, and banana was oriental fruit fly. So they were Bacteria dorsalis. So and also unidentified Bacteria history has only associated with papaya. So for this study, it will be needed to identify these unidentified fruit fly species. So this is about my research. So these are the selected references for my study, and thank you very much. As an agricultural country, Sri Lankan economy is filled with
Okay, thank you, Mr. Kavinda. That's also a very valuable research by diversifying the pest range of fruit flies. And the presentation is now open for the discussion, and I would like to invite our panel members to ask some questions or comments regarding the presentation.
Thank you, madam. Good afternoon, Mr. Kavinda. Mr. Kavinda, I have a small question. So, according to your results, it seems you analyze the relative abundance of each species in your traps, pheromone traps, right? So that methodology can be adopted for control of this fruit fly pest. So are there any evidences about how these different species and 2 different genera how can I say, response for different management practices? As example, how they respond for methyl eugenol or any other chemicals, whether we can use same chemical to manage those whole species or not.
Yes, sir, I got your point. Can you hear me, sir?
Yes, I can hear you.
Yes, sir. In my study, I didn't concentrate about the effect of these different chemicals, but there are findings about this each chemicals, how they affect to different fruit types. Fruit fly types.
Yes.
Basically, in Sri Lanka, the species we can see in our country, most of them are highly effective to the methyl eugenol. They have some affiliations for some. They are another chemical names then so they are also the effectiveness, but the species I got in this research in my research, all of these species are highly effective to the methyl eugenol. That's why I use the methyl eugenol for this research study.
Okay, Mr. Kavinda. Thank you very much.
Thanks, sir.
Thank you, madam.
Yes, madam, I can't hear you, madam.
Madam, we cannot hear you.
Thank you, Mr. Sirivardhana, for commenting on his presentation. And I have a very small question from you, Mr. Kavinda, that based on your findings, which fruit fly species should be prioritized to
Yes.
control?
Yes, madam. There are two fruit fly species to prioritize because in our country, Bacteria dorsalis and Bacteria candiensis are main considered at high abundant and highly effective fruit fly species for all the fruit flies, and also they have high pest range. So these two fruit flies are the main should be the main concern for the control. So the two species were the Bacteria dorsalis and the Bacteria candiensis.
Okay, thank you. Thank you for your explanations, and thank you the panel members. And we would like to move next presentation. And the next research is optimizing post-transplant recovery and yield of rice using different compost in modified depot nurseries. The presenter is Mr. HMASB Jayasinghe. I would like to invite Mr. HMASB Jayasinghe to present his valuable findings with us.
Good afternoon, everybody. I am HMASB Jayasinghe, the presenting author of the study entitled with Optimizing Post-Transplant Recovery and Yield of Rice or Rice as sativa using different composts in modified depot nurseries. Rice feeds over half of the global population. To meet the growing demand, we need to achieve 1.2 annual production increment. Transplanting improves planting efficiency and weed control, but introduces a major drawback: transplanting shock, a delay in seedling recovery. So, how can we reduce this shock? Transplanting shock delays growth, reduces tillering, and affects the yield. Machine transplanting especially needs 2-3 extra days for recovery. So our goal was to test whether different composts in depot nurseries can mitigate transplanting shock and boost the yield. We prepared modified depot nurseries according to the knowledge bank of International Rice Research Institute and tested 4 composts: kitchen waste, soybean residue, alfalfa and commercial compost. The variety was Japonica rice Nippon bere. Let's look at how we set up the trial. Seedlings were grown for 14 days and then transplanted pots. We used a randomized design with 5 replicates per treatment. Evaluations were done at 4, 8 and 12 days after transplanting on 4 indicators: plant height, green leaf number, spread value and hydrogen peroxide concentration. Also, we recorded the days to heading and grain filling percentage. Let's move to the results. At 4, 8 and 12 days after the transplanting, seedlings grown in alfalfa compost showed the highest green leaf number and plant height, indicating better early growth and recovery. This reflects reduced transplanting stress. The spread value, which indicate chlorophyll content and the potential of photosynthesis, were also highest in alfalfa treated seedlings. In contrast, hydrogen peroxide associated with the oxidative stress was lowest in alfalfa treatment. This suggests these seedlings experienced less transplanting shock and resumed growth more quickly. When considered the heading date, the earliest heading date was recorded from alfalfa treated plants, which indicates alfalfa treated plants reach their maturity earlier. Meanwhile, the highest field grain percentage was also recorded from alfalfa treated plants. Hydrogen peroxide produced in plants in response to stress conditions. After the transplanting, plants experienced water stress due to root system damage. However, A low hydrogen peroxide concentration and fast recovery have been observed from alfalfa treated plants. How this happened? Because alfalfa contain tricontanol, which is a natural plant growth promoting substance. It's looking alfalfa treatment helped seedlings cope with stress and establish quickly during stress condition. As a result, alfalfa treated plants had better early growth, earlier heading and higher field grain percentages. To conclude, alfalfa compost significantly reduced transplanting shock, promoted early vigor, and improved yield indicators. This suggests its promising role in sustainable rice farming. However, more trials with different rice varieties are needed to confirm these results. Our experiment was conducted based on the previous studies mentioned in the reference list. We would like to acknowledge Faculty of Graduate Studies, University of Ruhuna, Sri Lanka, Agricultural Research Station Kagawa, Japan, and ISATRS 2025, University of Colombo, Sri Lanka. Thank you.
Okay, thank you, Mr. Jaisingha, for your research findings. And it was actually address critical challenge in rice cultivation, that is a transplant in shock. And so I would like to invite our panel members to ask questions or give some comments to the presenter.
Okay, thank you. In your study, you use one rice variety. So is there any special reason to select this variety?
Sorry, madam, I didn't hear you.
Is there any special reason to select this variety for the study?
Yeah, actually, madam, this study was conducted in Japan. So in Japan, Nippon berry is a main rice variety, and also Nippon berry considered as a model rice plant, which commonly used in many rice experiments, especially even for the rice genome sequence analysis. Also, this variety has been
Can you hear us?
yeah. I can hear you.
Okay, right. My next question is that you collect the data on the parameter for hydrogen peroxide concentration. Okay.
Yeah.
Then can you explain why you get that parameter and how you get it during your study period?
Hydrogen peroxide is produced in plants in response to stress conditions, especially oxidative stress. So, under the condition of transplanting, so plants face to stress conditions because of the, as I mentioned in the presentation, due to the root system damage. Even though the root system damage, transpiration is continually happens. So plants experience face to water stress conditions. Under that conditions, to survive the plants, the hydrogen peroxide is produced. So when I consider I continue to the how I did that, actually it's a spectrophotometric analysis. So if I explain the methodology briefly, it's firstly we have to grind the leaf samples of
rice plants as we do during the DNA extraction, and we have to treat the homogenized solution with phosphorus buffer followed by the chemical called PVP, polyvinyl prolipyrene. Then after that, we have to do centrifuge under the 16,000 RPM for 10 minutes, then followed by the spectrophotomic analysis. By that, we can evaluate the hydrogen peroxide concentration.
Okay. Okay, thank you.
Thank you very much.
Thank you, Mr. Sorry, Jaisingh? Yes.
Yeah.
Can
Sir?
I hear you? Yes.
Yeah, I can hear you.
Mr. Jaisingh, this is practical question about your research. So you have used compost media for the layer as a modified dapple nurseries. So
Yes.
after modified dapple nursery, normally we use these nursery plants for machine transplanting. So with using different compost mixtures, according to your observation, this is not just analysis. I ask, according to your observation, how it affect on root growth, whether it enhance root growth or how it affect on machine transplanting. Just on
Actually,
your observation.
Yeah. Even within the different compost media I have been used, I have observed different root growths. For example, especially when compared with the direct seeding, so all modified dapple nurseries had higher root growth. So because I think it may be because of the direct seeding plants have plenty of water, but in case of modified dapple nurseries, they don't have such plenty of water. Just we are watering twice a day, so they have to overcome that challenge. So they have to develop their root system. So in that case, I have observed the root development is much better in modified dapple nurseries.
So this is my small suggestion. Next time you can identify how it affect. Just you can observe how it affect on because after we use compost mixture, sometimes it may affect on machine transplanting. It may difficult to transplanting and detach the planting from the media. So it is better to observe that also.
Yeah. Thank you, sir.
Thank you.
Okay, thank you, Mr. Jaisingh, for sharing with us your findings and explanations given. And next, we will move to the presentation of optimization of carbon to nitrogen ratios and assessing salinity tolerance in Metarhizium species for biofertilizer development in Vigna and unguiculate. And the presenter is Ms. Pereira. I would like to invite Ms. Pereira to present her valuable findings with us.
I will be presenting my research on optimization of carbon to nitrogen ratios and assessing salinity tolerance in Metarhizium for biofertilizer development in Vigna unguiculata cultivation. Agriculture today faces immense pressure due to factors like population growth, climatic change, urbanization, phytopathogens, and abiotic stressors such as soil salinity. With rising global demand, we need to cultivate high quality crops with maximum efficiency and minimal waste. However, conventional farming heavily relies on chemical fertilizers, while effective for pest control and boosting yield, cause detrimental environmental consequences. Therefore, agriculture practices must adopt eco-friendly alternatives like biofertilizers. Among these, metarhizium is a potent biocontrol agent and beneficial plant symbiont that promote plant growth and development through root colonization, improved nutrient uptake, and biological nitrogen fixation while enhancing stress tolerance and immune response. However, its effectiveness depends on nutrient availability, especially carbon and nitrogen. The carbon to nitrogen ratio in growth medium is key determinant affecting mycelial growth, sporulation and virulence. By optimizing media composition through controlled CN ratio and balanced mineral supplementation, we can maximize fungal biomass and conidial viability in agricultural and environmental application. Salinity has become a major environmental concern as most agricultural plants are sensitive to salt. Excessive salt accumulation in the soil disrupts plant growth through osmotic stress and ionic toxicity. For metarrhizome to be effective in such conditions, we need to understand its salt tolerance and how nutrient ratios affect its performance under saline stress. Vigna unguiculata was selected as the model crop due to its global importance, high nutritional value, adaptability and resilience. However, its production is often challenged by both biotic and abiotic stresses which significantly affect plant establishment, population growth and yield potential. By fostering a healthier soil microbiome, biofertilizer-based metarrhizium can improve soil structure, enhance microbial diversity, and increase nutrient availability. For media optimization, glucose and yeast extract were used as carbon and nitrogen sources respectively. The carbon concentration and CN ratio were calculated based on the carbon and nitrogen content of glucose and yeast extract. During the CN ratio studies, the carbon concentration was maintained at a fixed level while CN ratio was adjusted by varying the yeast extract concentrations. 3 carbon concentration and 4 CN ratio were tested. The growth was measured by colony diameter, sporulation and biomass production. For salt tolerance testing, I selected the media compositions yielding higher sporulation rates from the CN optimization which were all at the 20:1 ratio and tested them under various salinity levels using sodium chloride addition. Seed germination assay was also conducted. Results showed 8 gram per liter carbon concentration was appeared to be near the optimal for growth and sporulation with no significant improvement beyond this point, indicating carbon was no longer the limiting factor. The 20:1 CN ratio consistently resulted in the highest colony diameter, sporulation and biomass production, indicating that a lower natural limitation favors development, while 160:1 ratio was declined growth and spore counts dramatically due to severe natural restriction. For salinity, biomass production was best under non-saline conditions. But interestingly, 6 gram per liter carbon with moderate salinity actually promoted growth, suggesting stress response mechanisms like osmoregulation. The fungus remained resilient at 4 to 6 decisiemens per meter salinity levels at moderate carbon concentration, indicating a critical threshold where metabolic adjustment helps sustain growth under saline conditions. Most importantly, seed coated with metarrhizium spores grown at 8 gram per liter carbon concentration showed the most pronounced improvements in coffee seedling growth and vigor. This optimal concentration enhanced both germination and early stage plant development, while 6 gram per liter showed moderate benefits and 10 gram per liter actually showed slight decline, possibly due to metabolic stress. In conclusion, this research identified that 8 gram per liter carbon concentration combined with a 20:1 CN ratio optimally maximized metarrhizium growth, sporulation and plant growth promotion. These findings demonstrate that optimizing nutrient condition for metarrhizium can significantly contribute to sustainable agricultural practices by reducing reliance on chemical inputs while improving crop resilience, especially under stress conditions like salinity. Thank you so much.
Yeah, thank you, Ms. Pereira, for the valuable presentation and valuable findings. And the study highlighting the importance of CN ratio in maximizing fungal growth. And I would like to invite our panel members to comments and give some valuable comments and questions, ask questions from the presenter.
Thank you, Ms. Pereira, for your interesting presentation. So, you mentioned that this fungi species, Metracillium, Metahericium, ne? Yes, Metarhizium species. So, you did a laboratory experiment to clarify what CN ratio is suitable for better growth of this Metarhizium species.
Yes, sir.
So, how can we practically apply this research findings on our agriculture field? Whether you are going to apply these spores as a seed coating materials, or you are going to apply this fungi species on composting, or like can you please explain me how we can practically apply this knowledge?
Thank you for the question, sir. Actually, when identifying the best optimization media for metarhizium, we can produce spores. Then we can use them for the formulation of biofertilizers, which can increase the plant growth and plant nutrient uptake. Because the primary scope of this study is the product development, which should be cost effective and the local available resources used. Based on this concept, where I worked at the biofertilizer manufacturing company, so they encourage us to formulate a biofertilizer which based biofertilizer based metarhizium based biofertilizer which can be used for the legumes because legumes are really nutritional valuable, and there is a high demand for because demand in the agriculture field. So I want to make a foundation when what should be the best CN ratio for metarhizium. Based on that, metarhizium can produce highest amount of spores using that nutrition condition. So we can use that findings for the further development of biofertilizer based on metarhizium. So that should my answers.
Thank you. I can understand. So, yes, I wanted to understand, clarify whether how use these findings. Then I can understand. First, we should grow this fungi in the laboratory. Then we can inoculate it into the biofertilizer as an inoculum. It's okay. Okay. Thank you. Thank you very much, Ms. Pereira.
Yeah, thank you, Mr. Srivardhana, for commenting on her presentation. And also, I have a general comment that you did that experiment in the laboratory conditions, and Have you planned to do any field trials because you optimized one biofertilizer? And do you have any plans to apply it in the field?
Yes, madam. I basically did the development and lab validation, but I plan to do field trials as future work.
Okay, thank you. And it's very valuable research. And applications in fields, that's very important. So we are also recommending to do that in the field trials. Okay, thank you. And we will move to the next research. And that is quality assessment of Sri Lankan banana varieties at different ripening stages. The presenter is Ms. P.D.Y. Premaratna.
Hello everyone, I am Dilashya Shodha from Department of Food Science and Technology, Faculty of Livestock, Fisheries and Nutrition, Wambi University of Sri Lanka. I am presenting my research quality assessment of Sri Lankan banana varieties at different ripening stages. Banana are a staple fruit in globally, but their physiochemical, nutritional, functional property vary across varieties and ripening stages, influencing their dietary value and industrial application. In this study, I analyzed the 8 banana varieties with 3 different ripening stages. As the objective, mainly I comparatively analyzed the approximate mineral composition, physiochemical and functional properties of these banana varieties at different ripening stages. These are the banana varieties that I am analyzing in this study. When selecting the ripening stages, I am selecting the more green than yellow color stage, more yellow than green color stage, and the fully yellow color stage. As the methodology, first I am collected unripe banana sample from Suryava, Ratnapura and Kurunagal areas. Then sample were washed with clean water and stored on the same temperature, stored under room temperature condition for the ripening purposes. Then I dried banana powder, grind, sieve and got dried banana powder for further analysis. As approximate composition, moisture, crude fat, crude fiber, crude protein and moisture content were analyzed. As the physiochemical properties, pH level, titrable acidity, total soluble solid and pulp-to-yield ratio were analyzed. As the mineral composition, I analyze sodium, potassium and magnesium using the atomic absorption spectroscopy. As the functional properties, I analyze the total phenolic content, total flavonoid content and the antioxidant activity. To analyze the functional property, I get the dried banana powder extraction with the methanol using the sonication method. Data analyzing using SPSS software. First, I analyze the data according to the banana varieties at different ripening stages. Then I analyze R3 ripening stages among the varieties. As the resultant discussion, physiochemical properties were analyzed. Pulp-to-peel ratio high in Ambul and Suandal banana out of other varieties. Consider the total soluble solid content. Total soluble solid content increase R1 to R3. pH value and titrable acidity. pH increase with the ripening process while decrease in the titrable acidity. Consider about the moisture content. Moisture content typically increase with the ripening process, but Ambul and Ratkehel, they did not show any differences among the ripening stages. Considering about the ash content, ash content mainly decrease with the ripening process, but in Suvan there is no significant difference in the R1 to R3. Crude protein content also decrease with the ripening process, but in Ambul Kehel there is no significant difference among the R2 and R3 varieties. Crude fiber content also reduced in both varieties from R1 to R3. Considering about the crude fat content also reduced among the R1 to R3 among banana varieties, but in Ratkehel there is no significant difference in crude fat content from R1 to R3. Considering about the functional properties, total phenolic content, total flavonoid content, and antioxidant activity, all three parameters significantly high in R2 stage than R1 and R3. This is one of the main thing that I found in this research, so that we can tell R2 stage more functionality suitable than other R1 and R2 in all banana varieties. When considering about the mineral content, potassium content, magnesium content, and sodium content, potassium and sodium content did not significantly differ from R1 to R3, but magnesium content significantly reduced from R1 to R3. Then I compare the fully yellow ripening stages among banana varieties. When considered about the proximate composition, moisture content high in Ambu, Kolikuttu and Netri Palam banana. So show the highest crude fiber content than other varieties. Crude protein content also high in Ambu and Kolikuttu. Total ash content also high in Ambu and Kolikuttu banana than other varieties. Considering about the crude fat content, out of these 8 varieties, Netrapalam show the highest crude fat content. But crude fat content is significantly low all these varieties. As the physiochemical properties, highest pulp to pale ratio showing in the Sundar cases, and Kolikuttu and Ratkehel show the highest pH value, while tritriple acidity show lowest in the Ambul-Kehl. Total soluble solid also high in Kolikuttu banana. Considering about the functional property, total phenolic content high in Netrapalam banana, total flavonoid content high in Kolikuttu banana, and both Ratkehel and Netrapalam show the highest antioxidant capacity. Out of these, at R3 stage, Suandal, Kolikuttu and Ambu had the best value across most proximate functional parameters. This makes them ideal for the value-added food product or nutritional uses. As the conclusion, banana variety and ripening stage significantly affect the quality of the banana. So the Kolikuttu and Ambu show the best nutritional and functional profile out of these varieties. R2 ripening stage had the highest antioxidant activity than other. This finding can help promote underutilized banana varieties in both food processing and dietary deification. I would like to thank my supervisor, Ms. Adhikari, from Department of Food Science and Technology, Walmer University of Sri Lanka. And I would like to thank all others who helped me to complete my research in success. Thank you.
Thank you, Ms. Premaratna, for your nice presentation and valuable findings. And the study highlighting that how ripening affect the nutritional and functional quality in Sri Lankan banana varieties. And I would like to invite our panel members to comment on the research findings and ask some questions.
Okay. Miss, thank you for your presentation. Yes, Ms. Premaratna. Then, according to your clarification that you categorize the different ripening stages as R1, R2, R3, right? Is it based on only the peel color?
Yes, it's only based on the peel color.
Okay, then according to you are finding that in R2 stage, R2 ripening stage, is it what is its palatability level? It is edible.
Yes, it's edible. Mainly
Okay.
in R2 stage, it's more yellow than green color. In that stage, we can eat that stage also.
And R2, R3, both are possible to
Yes.
eat.
Yes.
Right. Okay. Then according to your findings, right, in which variety or which type of banana was selected and in which ripening stage the best as the best?
According to the finding, we can't conclude that one variety is best. According to the finding, in some health conditions, doctors recommended some fruit don't eat like that. So that according to the mineral content is ample show the higher potassium content in the ripening stage. So that doctors don't recommend to get banana. It's maybe not good for like kidney patients like that. So that when consider about the functional property, R2 stage more better than any other stage. Other stage and Netrapalam, Kolikuttu and Ratkeher. Considering the total phenolic, total flavonoid and antioxidant, they have the highest value than others. So we can conclude that antioxidant capacity high in total phenolic R2 stage total phenolic content high in Netrapalam, something like that.
Okay, that mean when consider about different property that results are showing the different varieties of the
Yes,
types of banana, and the ripening stage also the same, no?
Yes, madam.
Right. Okay. Thank you.
Thank you, madam.
Okay. Thank you, Ms. Premaratna, for your valuable findings share with us and the explanations. Next, we will move to
Thank you,
the next presenter. Okay, that is production of single cell protein from banana peels, a sustainable approach to protein enriched biscuit. The presenter is Mr. Ratnayaka. I would like to invite Mr. Ratnayaka.
Hello everyone, today I will be presenting my research on production of single cell protein from banana peels, a sustainable approach to protein enriched biscuits. With the world facing rising protein demand and growing environmental concerns, it is vital to find sustainable food sources. At the same time, a large amount of fruit waste, especially banana peels, is discarded each year. In Sri Lanka, over 35 million tons of banana peel waste are generated annually. Single-cell protein refers to dried microbial biomass that can be used as a low-cost alternative protein source. This research explores the use of banana peels, especially Ambul and Sini varieties, as a substrate for single-cell protein production using Saccharomyces cerevisiae. The general objectives of the research is to produce single-cell protein using banana peels via submerged fermentation. The specific objectives are to determine the most suitable banana peel variety for single-cell protein production, to evaluate the physical and physiochemical properties of single-cell protein, to incorporate single-cell protein into biscuits, evaluate its impact on nutritional content, and determine the shelf life of the protein-enriched biscuits. This is the methodology followed throughout the research. First one is the sample collection and preparation. For this, banana peels were collected, cleaned and dried and ground into powder. Then after that, the proximate analysis tests were conducted for the two banana peel varieties to select the most suitable banana peel variety for single cell protein production. Then the dried banana peel powder was pre-treated and hydrolyzed using HCl and fermented it with Saccharomyces cerevisiae. Then the single cell protein was harvested and dried. After that, the harvested single cell protein was analyzed using some physical and physiochemical analyzing tests. Then single cell protein was incorporated into biscuits at various concentrations, 5%, 7.5% and 10%. Then the nutrition analysis and sensory evaluation tests were conducted. After that, the accelerated shelf life test was conducted for the prepared biscuit samples. Now moving on to the most important part, the result section. I first compared the proximate composition of the amber and seamy banana peels. Based on the highest nutrient content, particularly crude protein and carbohydrates, amber banana peel was selected for single cell protein production. Using 1 kilogram of amber banana, I obtained around 11.05 gram of single cell protein powder through submerged fermentation. The physical analysis of the single cell protein was promising. Dry cell weight was 11.05 gram. The protein yield was 31.2%, showing efficient biomass conversion. Water absorption capacity was 1.8 milliliter per gram, indicating the single cell protein can retain moisture well. Bulk density was 0.5 gram per cubic centimeter, suggesting good flow properties for food application. When considering the physiochemical analysis test, the produced single cell protein contained about 24.2% crude protein along with the acceptable levels of moisture, ash, fat and fiber, confirming its value as a food-grade protein source. I then tested single cell protein incorporation into biscuits at 5%, 7.5% and 10% levels. Through sensory evaluation result, the 5% single cell protein biscuit received the highest preference in terms of taste, texture, color and overall acceptability. Nutritionally, this 5% single cell protein biscuit had increased protein content compared to the control, demonstrating that single cell protein can improve the nutritional quality of everyday food products. Lastly, I assessed the shelf life of the biscuits using accurate storage conditions at 25 Celsius, 35 Celsius and 45 Celsius. I tracked moisture content over 4 weeks. The best storage condition was 25 Celsius and the estimated shelf life was around 10 weeks, as the rate of moisture absorption and therefore spoilage was lowest at that temperature. In conclusion, this study shows that banana peels, particularly from the amber variety, are an excellent raw material for producing single-cell protein. The protein-rich single-cell protein was successfully used to enhance the nutritional value of biscuits without compromising the taste. This approach not only reduces the fruit waste but also offers a low-cost, eco-friendly protein source. These are some references used for this research, and thank you very much for your attention.
Thank you. And now the presentation is open for the discussion. I would like to invite panel members to ask some questions and give some comments.
Okay, Ms. Ratnayaka, thank you very much for your presentation. And I think this is an interested study, so that I would like to know that what is the that biscuit amount, or I don't know whether you have analyzed this as suitable for this, right? So, but what is the amount of biscuit should be consumed per day to achieve to reach the protein requirement, to get the daily protein requirement? Ms. Ratnayaka, can you hear us? Ms. Ratnayaka, can you hear me? We cannot hear you. I think your mic is muted. Please unmute your microphone. Ms. Ratnayaka? Okay, Ms. Ratnayaka, are you there?
Hello, Miss.
Okay, I think
Can
now
you
you
hear
can
me?
hear. Yes, yes, we can hear you well. So, my question is that can we achieve the daily?
Yes, Miss.
Okay, can we achieve the daily protein requirement using these biscuits? And
Yes, Miss. In my according to my results, there is a amount 10% protein percentage in that biscuits, but we can achieve more protein percentage level by improving the single cell protein percentage in the biscuits. We can do this by using optimization the fermentation conditions, and use more single cell protein for the biscuits by applying more percentage. We can get the more protein percentages.
okay, during your study, that what are the challenges you have faced while conducting the study? Ms. Ratnayaka, can you hear us?
In my study, madam, I faced
Okay, that because of some network issue actually that we cannot do well.
Fermentation conditions like we need to get the I can hear you. Can you hear me?
Okay, thank you, Ms. Ratnayaka.
Okay, thank you, Mr. Ratnayaka, for your sharing with us your findings and your valuable research. And we will move to the next presenter and the next research findings. That is pollination, negative effect of noise pollution on insect pollinators. The presenter is Mr. D.S.B. Ratnayaka. I would like to invite
Hello, everyone.
Mr. Ratnayaka to present his valuable findings with us.
Hello everyone, I am Tuminda Sanjay Bandara Ratnayaka from Industrial Engineering Training Institute, National Apprentice and Industry Training Authority, Katubedda, Sri Lanka. My research title is Pollination, Negative Effects of Noise Pollution on Insect Pollinators. So then let's go to the introduction of my research. Actually, majority of global crops are relying on pollinators to maintain the food security. Approximately 75 to 90% of flowering plants are dependent on biotic factors such as bees, butterflies and flies rather than abiotic carriers such as wind. Actually, insect predators are playing a vital role in plant reproduction. Actually, it makes the establishment of broader ecosystem processes where that contribution. With the time, alarming declines can be seen affected on the pollinator populations. I think it is reported from various parts of the world, posing a serious ecological threat. When we are considering about the various factors relying contributed for the declining of pollinator population, noise pollution is very important. In here, we are discussing about how noise pollution affect on the decline of insect pollinators. Okay, let's go to the objectives. First one is identification of negative effects of noise pollution on insect pollinators. Second one is identification of the various measures to reduce the negative effects on the pollinators. This is my methodology. This is a qualitative research methodology. This systematic literature review was conducted by me from referring the 2021 to 2024 international and national sources. Scholarly articles, books, dissertations and conference papers across various disciplines I examine, especially agriculture, entomology and environmental sciences. It makes the comprehensive advanced understanding of the issue. I use various search engines and databases to gather various diverse viewpoints and data sets. This is my research and discussion. What are the negative effects of noise pollution on insect pollinators? Actually, first one is disrupted reproductive behavior, very important factors. Reproduction patterns are disturbed by that factors. Next thing is impact the communication. Communication will be collapsed. Next thing is physical stress on the insect pollinators and weak immune responses on their health condition and altered pollination patterns, very serious factor. Then what are the various measures to reduce the negative effects? Actually, urgent need for the long-term ecological studies is particularly focusing on species like bumblebees are very important. Actually, various research should be conducted to by targeting various types of pollinators to conserve, especially based on the pollinators who are scattered in urban environment. Actually, we will be contributors in imposing of various types of institutional regulations. We will be able to get the support of both public and private organizations to implement that things to reduce the impacts on noise pollution. These are my conclusions of the research. Given their dispensable role in global food system, actually protecting the pollinators must be prioritized. Very important thing. Future research should be focused on policy-driven conservation initiatives that safeguard insect pollinators while we should protecting the sustainable agriculture practices. These are my references. Pollinator diversity benefits, natural and agriculture ecosystems, Katumo. Then impacts on multiple pollinator activity on road verges. Exploring noise pollution, cause, effects and mitigation strategies. Anthropogenic noise affects insect and arachnid behavior, thus changing interaction between species. Next thing is opportunities and threats for pollinator conservation, like that. These are my references. Thank you very much for the contribution of my.
Okay, thank you, Mr. Ratnayaka. That is very good research approach, because these days is in terms of pollination, that insect pollinators are that reducing rapidly, and you have taken an approach to find out what are the effect of noise for these reducing that insect pollinators. You have done a literature survey that I think better to expand that research to another level and finding what are the solution you can suggest for that overcome this problem. Okay, now the presentation is open for the discussion, and I would like to invite panel members to ask some questions and give some comments regarding the research.
Yes, Mr. Duminder. As you mentioned, these days the hot topic is that bee how effect on this noise, as well as mobile signal on bees as well. So it's very interesting. So did you identify any possible methods, strategies to rehabilitate this effect, the already affected effect by noise on insect community? How we can overcome? What are the possible strategies we can adopt to reduce or overcome this problem?
Thank you, sir. Can you hear me?
Yes, Mr. Duminda, we can hear you.
The thing is, actually, under Sri Lankan conditions, there are no any specific conservation method for specialized for the conservation of insect population. The thing is, actually, think about the any other wildlife animals. There is a separate department called wildlife conservation and any other things, but there are no any specific organizations or any other specific government establishment to conserve the pollinators, like a very important group of insects. Actually, the most important thing is under Sri Lankan conditions, we are doing some research, and sometimes we are non-government organizations and some environmental organizations are telling about the importance of the conservation of that type of insects, but no, local populations are not following that type of method. The thing is, actually already we have identified that some type of noise pollution is directly affecting on the pollination behavior, reproductive behavior, sometimes immune response, and then alternative pollination patterns, like that. So then we are talking about that things under certain conditions, but we are not practically, we are not applying how to conserve that type of insects, because we are not conserving them. Because the thing is, actually there's a direct influence on the food security. Okay, we are concerning about the final then end product. What is the reason for the actually suppressing of the yield of that crop? But we are not concerning about the what are the causal agent, who is the responsible pioneer for the pollination process of that things. I think this is the normal nature. I think from our university levels, not from the university levels, in our school levels, at first we should establish specific attitude improvement, the attitudes of the students, and after that we should convey that attitudes for the university students, that while we are establishing that concepts, we should disperse that for the overall society. Then specific thing is, I mentioned about the bumblebee. Actually, bumblebee is a very, very important type of insect,
because there's a specific pollination method called bus pollution, because they are then absorbing the pollen from the anther, and they are dispersing the anthers for any other plants, because these types of insects should be introduced under Sri Lankan conditions. Actually, then why already then vanilla and any other plants are actually they are lost with their pollinators, because we should do under artificial manner. Think about the carpenter beetle, because actually there's a practical situation with Bachelor of Science in Agriculture or any other agriculture programs. We are making insect box. For the insect box, we are killing number of carpenter beetles for the specimen collection. In my undergraduate level, plant protection technology, our batchmates, I think 130 batchmates are there, because 130 bumblebees are killing. So then we are preserving for our academic purposes, but in fashion fruit, I think bumblebee specified for fashion fruit cultivation. What's the way of pollinating the passion fruit surrounding our environment? These are the practical issues, sir. Actually, I don't know whether my opinion is okay or not. Then we should, in government or any other non-government party should involve for the conservation of pollinator population than that, I think.
Okay, thank you, Mr. Duminda. Since we have time limitation, thank you for your nice explanation, and I wish you to extend your research beyond this literature survey up to the field level research. Thank you.
Thank you very much, sir. Thank you very much, madam. Okay, thank you.
Okay, thank you for fruitful discussion and your explanations. And now we will move to the next presentation and next research findings. The topic is selection of suitable rootstock for cucumber cultivation grown under protected house condition. The presenter is Mr. K.K. Heen Kender. I would like to invite Mr. K.K. Heen Kender to present his valuable findings with us.
Hello everyone, I am Kalindukushan Hinkende. Here present research finding on selection of suitable rootstock for cucumber cultivation grow under protected house condition. This is the content of my presentation. Let's move to the introduction. Cucumber is the widely cultivated, economically important vegetable in Sri Lanka. Recently, cucumber cultivation under protected condition gained popularity due to high productivity and the year around farm. However, but it has also led to challenge primary soil borne disease. Grafting is the effective method in horticulture and it can be used for protected cucumber cultivation. Cucumber grafting is the novel technique used normally cucumbers scion made with the compatible and the vigorous rootstock. This method improve resistance to soil borne pathogen. Grafted plant normally show better growth and high yield compared to the other non-grafting plant. Let's see the problem statement. Root pest and disease affecting to the seedling is a major problem in protected cucumber cultivation. Grafting is the new practice used for the producing planting material for vegetative propagation of cucumber for protected cultivation. Therefore, important to identify and select appropriate suitable rootstock for cucumber grafting. Let's see the objective of this study to select of suitable rootstock for grafting. Let's see materials and method. This experiment was conducted at the Regional Agriculture Research and Development Center, Bandarawila, and the experiment was organized as a complete randomization design with the 5 replicate. Each replicate consists of 10 pots. So let's see the treatment of this study. There was 5 treatment for rootstock and which cucumber scion, buttergourd, pumpkin, ash pumpkin, kakiri rootstock used for this study. Treatment 5, it is the cucumber without grafting as the control. In Establishment of scion and rootstock process we prepared both rootstock and scion to seed collection, treatment and nursery practice. After that, healthy plant select for whole insulation grafting used for this grafting method. Then healing, acclimatization and process was done. Then data collection was done. Under the data collection, firstly successful rate cucumber grafting was initially evaluated. After that, from the successful grafted plant height, number of the used per plant, total wet and dry weight of the plant, wet and dry weight of the root, and also maximum root length was measured. After that, collected data analyzed using the Minitab statistical software. Now I am talking about the result when given this research. Let's see the success of grafting. According to success percentage, treatment 1, treatment 2 and treatment 5 show success of the grafting. Treatment 3, it means the ash pumpkin 1 get only 17% percentage success, and treatment 4 get 0% success. It means not success. Then after treatment 3, plant also wilted and dried before the data collection. Therefore, the data collection is to continue only using treatment 1, treatment 2 and treatment 5. Plant height significantly higher plant height recorded from the treatment 1. Let's see number of the true leaves. You can see here there is no significant difference between number of the true leaves. Let's see maximum root length according to the result. Significantly higher root length was recorded from treatment 1. Fresh weight of the plant significantly higher plant fresh weight recorded from the treatment 1. Let's see the fresh weight of root. There was not significantly different between treatment 1 and treatment 2, but significantly different compared to the control. Fresh weight of the shoot, significantly higher fresh weight of shoot recorded from the treatment 1. Root dry weight, there was no significantly difference between treatment 1 and treatment 2, but compared to the control, treatment 1 and treatment 2 significantly high. Shoot dry weight significantly higher treatment 1. Plant dry weight significantly higher treatment 1. Conclusion, according to the result, treatment 1 it means butter gourd with cucumber scion show significantly higher value for the treatment in the root length, plant weight, shoot fresh weight, shoot dry weight, plant dry weight compared to the other treatment, and also treatment 1 show significantly different compared to the control in the root fresh weight and root dry weight. So we can conclude that the butter gourd is the suitable rootstock for successful grafting of cucumber. As way forward, further study are needed to confirm the finding, and also study should be needed to identify the field performance of the grafted plant. This is the reference. My presentation is over. Thank you.
Okay, thank you, Mr. Heenkender, for your nice presentation and presenting your valuable findings with us. And now the presentation is open for the discussion, and I would like to invite panel members to comment on this and give some comments and ask some questions regarding the findings.
Okay, Mr. Hinkender, that in your research, you have selected different plant varieties, no? Species, 5 species as root stock, no?
Yes, ma'am.
Okay, then when considering the grafting, so how you select the maturity stage of the rootstock? What are the factors you considered when selecting the grafting stage of each root stock? Is it same or different? If there is different, what are the different factors?
Actually, I select local seeds. Reason is local seeds have good environment, bad condition resistant and pest resistant. I think consider root length and root system about rootstock. Hello.
What are the parameter or the
Hello.
You considered there? Stem girth.
Hello, madam. I cannot hear you.
Stem girth for the when selecting the maturity stage of rootstock for the grafting.
Hello.
Mr. Indian, I think the problem in your side, we can hear the panel members' voice.
Okay.
Hello.
Can you hear us now?
Yes, ma'am.
Okay. That you finally recommend bottle gourd as the good rootstock, no?
Yes.
Okay. Then have you that applied that finding in the that farmers field level or protected cultivations?
No, madam. I hope to continue this study future to further investigate the field performance of the grafted cucumber plant after the field plant.
Okay. Okay, Mr. Chatura, do you have any questions?
And we can move forward. Thank you.
Okay, I think.
It's okay. Thank you, Mr. Hinkender, for your explanations. And now we will move to the final presentation of this session and final research findings. That is assessment of growth and yield traits of novel mushroom varieties for commercial cultivation. The presenter is Miss P.B.D. Darshika. I would like to invite Miss Darshika to present her valuable findings with us.
Good morning, all of you. I'm Darshika Jayavante, and today I'm going to present you about our research work on assessment of growth and yield traits of novel mushroom strains for commercial cultivation. Mushroom, it is a eukaryotic macrofungus, and in current time, there are about 200 mushroom strains we can use as foods. Unfortunately, only 35 strains of among them are currently being cultivated under commercial scale. In Sri Lanka, we also have a huge potential toward mushroom cultivation, but unfortunately, the commercial mushroom cultivation industry in Sri Lanka is remain underdeveloped due to many reasons, and among many, one is reliance of narrow genetic pool of commonly used strains. However, in global scale, there have been taken several steps to develop novel mushroom strains using conventional breeding techniques and modern biotechnological tools. Unfortunately, in Sri Lanka, we don't have such advanced method, and also the novel strains that we have developed are not being evaluated in systematic manner. So, to address these challenges, this study aims to conduct a comparative evaluation of growth and yield traits of newly developed crossbreed mushroom varieties. So the objectives of my research are to evaluate the growth and yield performance of newly developed mushroom strains and to calculate the biological efficiency of newly developed mushroom strains. So this is the methodology, and let us go through in comprehensive manner. First, substrate was prepared, and then 950 gram of substrate mix was filled into a polypropylene bag, followed by sterilization and spoon establishment under uh, sterile conditions. And these are the 4 treatments. So basically here we have used 4 pure mushroom strains and 4 hybrid or crossbreed mushroom strains. Each treatment has 20 number of replicates. So these are the measured parameters. Time taken to complete the spawn run, time taken to initiate the fruiting, average stalk length, average cap diameter, and finally, biological efficiency of newly developed mushroom strains. Then, the data analysis of the data were carried using a Minitab statistical package in ANOVA procedure at 0.05 significance level. Now let us move with results and discussion part. First, let us consider spawn run rate. As you can see, the treatment effect has significantly contributed for the spawn run rate, and treatment A performs significantly better than the other treatment. So treatment 8 is a crossbreed of American oyster and China. When considering about the duration for first flushing, treatment 8 shows the lowest mean, and though treatment 1 and treatment 8 are significantly different from other treatment 8, which is a cross breed of American oyster and China, shows the lowest mean, whereas treatment 2, which is the abalone, shows the highest mean. When considering about the yield performances, treatment 6 results the highest flush yield, which is the crossbreed of China and abalone. And again, we compare with other treatments. Treatment 6 have highest and significant biological efficiency value also. And when consider about the morphological characteristics and the stalk length, there is no significant difference between treatment 1, 2, 3, 4, 5, and 8. However, there is a huge significant difference of treatment 6, which is a cross between China and abalone. And there is no significant difference in the mean cap diameter among the different treatments groups that we have tested. And the conclusion of my research are, among the tested strains, hybrid strain American oyster and China demonstrated the highest significant faster colonization rate. China abalone recorded a significant higher yield and significant higher biological efficiency than pure breeds or pure strains. Overall, these results demonstrate that hybridization, particularly involving with China strain, can enhance tree production traits, making certain hybrids highly suitable for commercial scale mushroom farming. However, future research should focus on evaluating these high-performing hybrids for their new nutritional content, shelf life, and resistance to contaminants in order to adapt them in commercial cultivation. So that is the end of my presentation. Thank you for your patience.
Okay, thank you, Ms. Darshika, for your nice presentation and valuable findings. And now the presentation is open for the discussion, and I would like to invite our panel members to discuss the research findings.
Okay, Ms. Darshika, thank you for your presentation. And I want to clarify something. And what is the biological importance of biological efficiency in evaluating the mushroom strains? Then in your study, you have you have discussed about the different mushroom strains. Okay, so that in case of that, what is the importance of biological efficiency in evaluating those strains, mushroom strains?
So biological efficiency means that how these mushroom strains can effectively turn the nutritional components in the substrate to their mass, to their body weight. So that is why we have to measure this biological efficiency to identify how they can efficiently transfer the nutrient contents in the substrate to their body mass.
Okay, that when measuring it, what are the parameters you have considered?
So the biological efficiency was measured from fresh weight of the harvested mushroom was divided from the dry weight of the substrate.
Okay, it is in that weekly taken parameter or that?
No, this research was taken into the harvest of the mushrooms was taken in three flushes. So in one flush we calculate, and so altogether there was 3 biological efficiency was calculated, and the mean value was mentioned in the final present.
Okay, in your the strains, mushroom strains that you have used for your study, which one has the highest biological efficiency?
So according to our results, the overall oyster and abalone mixture, this is the hybrid of oyster mushroom, American oyster, and abalone is express the highest biological efficiency turnover.
Okay, thank you, Ms. Darshika, for your explanation. Thank you.
I have a small question.
Yes.
Okay, my question is when we are concerned about the mushroom cultivation, minimum shelf life is very important factor. I think I suggest to conduct number of research studies to find out the effective enhancing the effective shelf life for mushroom, because sometimes we are buying mushroom from supermarket or any other place. I think although we are storing under refrigeration, I think only we can put it up to 2 or 3 days, but it's start to yellowing. What's the reason for that part? Actually,
Mainly it is due to the discontaminations. That is why I suggest in my presentation we have to evaluate the contamination effects of this mushroom before we are going to commercial cultivation.
Okay, thank you very much for a nice presentation.
Okay. Thank you, Ms. Darshika, and your valuable explanations. And in this presentation, we concluding our session. And actually, we discussed and shared research findings of 12 research. And it is very diversified topics and different areas we discussed. And actually, I would like to thank all presenters, those who shared with us their valuable findings. And I would like to thank our panel members, Mr. Sirivardhana and Ms. Nelka, for sharing their comments with our presenters. And all presenters also, I would like to thank being with us during this more than 2 hours period. And with this, I would like to conclude the session. And thank you very much. And also the Miss Aishmanti, those that session coordinator of this session, and she with us. She also with us during this 2.5 hour period. And thank you very much all. And I would like to conclude the session now. Thank you.
Thank you, Madam. Now we have come to the end of the technical session 2. I would like to extend my heartfelt thanks to our session chair, Dr. N.P. Vidana Patirana. Madam, your presence added immense value to the session. I also want to express my gratitude to our panel members, Ms. S.A.P. Nelka and Mr. A.J.M.C. M. Sirivardhana, for sharing their valuable thoughts and time with us. Lastly, I would like to thank all the presenters for sharing your findings and contribution to the truthful discussion. Please join us with the session 3 of Crop Science and Emerging Technologies for Sustainable Agriculture will be started in a short while. Please stay with us. Thank you.
Good afternoon. Welcome you all for the last session of the first International Symposium on Agrotechnology 2025. The technical session is titled under the theme of Science and Emerging Agriculture. It is honored me to invite Dr. E.M. Vimalasiri, Head, Department of Export Agriculture, Faculty of Agricultural Sciences, Sabaragam University of Sri Lanka, to chair this session. Dr. E.M. Vimalasiri is one of the great researchers in the field of crop modeling and climate and he has been awarded as the researcher in the Faculty of Agricultural Sciences, Sabaragam University of Sri Lanka, for 4 consecutive years since 2022. It is honored me to welcome you for this session, sir. Then I would like to invite panel members, Dr. Supun Bandusekara, lecturer, Department of Agrotechnology, UCIARS, and Mrs. Kasuniketi Piarachi, lecturer, Department of Agrotechnology, UCIARS. Dear sir, Dr. Iranga Vimalasiri, I warmly welcome you to chair this session and proceed. The session is over to you. Thank you, sir.
Good afternoon. Thank you very much, Shature. Thank you very much for the introduction. Without taking much time, we'll start the session. First, I wish you all good luck, and we'll see how we proceed. So we can start with the presentations. Shall we move to the
Hello everyone, I am Niranji Madhusika Somaratna. I am going to present my review presentation titled on Waste-Derived Filter Media for Sustainable Household Aquaponics. This is the content of my presentation. There are different emerging challenges in global food production due to the growing population, decreasing land and extreme weather conditions. With increased urbanization, there are some alterations in the food demand in cities. Therefore, the sustainable urban food production identified as the solution for environmental challenges, social needs and the economic welfare. One of that system is aquaponics, which combine both aquaculture and hydroponics together. It simultaneously fulfills the essential nutrition requirement for plant growth and at the same time manage the waste generated through the aquaculture system. As well as aquaponics support to reach some sustainable development goals such as zero hunger and enable the accessibility to food as it is reached to food security. These systems heavily depend on the maintaining optimal water quality within the system. So, filtering system is identified as served as the indispensable component in this aquaculture system. Biobores expanded to lava rock and gravel are some biological and mechanical filters commonly used in aquaponic systems. But due to some limitations of these materials such as high shipping cost, customs and logical cost, clogging problems, provide trapping some substances, less longevity, there are increasing interest on using alternative filter materials. So, the objectives of this review is to explore the potential of various waste materials that can be used as filter media in aquaponics and evaluate and compare the effectiveness of these materials in terms of performance indicators like nitrate conversion, ammonia removal, pH stability, cost effectiveness and durability. Then we move on to the methodology part. A comprehensive literature search was conducted via Google Scholar and ResearchGate, focusing on peer-reviewed articles published between past recent 10 years, using a specific keywords such as biofilters, mechanical filters, wastewater treatment, and aquaponic systems. 55 peer-reviewed articles were screened and selected studies were analyzed based on their methodology and conclusion, focused on waste-derived filter materials, positive and negative results of analyzed materials, advantages and limitations of the selected materials when compared with commercially available and currently used filter materials, and remaining research gaps on the study area was also focused. 32 articles were deemed relevant and findings from impactful journals were considered. These are the keywords and search strategies. As results, we identified there are two main categories of data which we can collect: qualitative data and quantitative data. Qualitative data include there are some special essential features which should essentially have in the filter media such as porosity, durability, maintenance, chemical properties, iron absorption and removal like properties. So, these properties categorized under the qualitative data. Then other one is quantitative data such as it measures the percentage of substances removed from the filter media. So these are the identified potential waste materials which can be used as filter media in aquaponic system. First one is ceramics, then activated carbon, wood chips, biochar, paddy husk, nylon net scraps, coconut husk, coconut shells, wood shells and charcoal, horns and bamboo. Among these materials like biochar, ceramics and activated carbon showed the superior filtering features, and waste materials such as paddy husk, nylon, net scraps and bamboo shows the less effective performances when got the overall. Finally, we can conclude that despite positive results with materials like ceramics, activated carbon and biochar in nitrate conversion, can removal, pH stability, durability and cost effectiveness, still there is significant research gap is remaining. This research gap include long-term performances of these filter materials, degradation, how these filter materials degrade with the time, maintenance, how easy they can maintain, how about the maintenance cost, the effect of microbial interaction, toxicity and chemical stability. Therefore, standardized studies are essential to address these gaps and enhance the practical applications of these materials. These are the references. This is the end of my presentation. Thank you for your attention.
Right. Thank you very much for your presentation, Ms. Somarathan. I have a practical question to ask. Among the waste materials you studied, which show the most promise in terms of filtration efficiency as well as the long-term stability?
Can you again ask the question, sir?
Hello, can you hear me?
Yeah.
Among the
Can
waste
you again
materials
ask
you
the
find,
question?
let's say in the publications, which show the most promise in terms of both filtration efficiency as well as the long-term stability?
I think activated carbon and ceramics, but not all of we cannot. Activated carbon also have some limitations. It is, I think, it is mixed. We cannot get exact 100% materials cannot filter. All the materials have even one limitation, but I think activated carbon is best one.
Okay. Any questions? Any questions from the audience or the panel members?
Okay, thank you very much, Ms. Somaratana, for your nice presentation. I have one question. Am I audible to you?
Yes, sir.
All right. So you have done this research based on the waste material and use as a media. So my simple question is, why you mainly focusing on aquaponic system by using these waste material? So you can use any other things as well. So what is the main reason why you mainly focusing on aquaponic systems? Do you see any kind of potential?
Yes, sir. This is review, but in my undergraduate research, I did this in practical. That's in my undergraduate research. I used ceramics, nylon, scrap, paddy husk like material. So after doing the research, according to the conclusion, I thought actually there is potential of these materials which we can use as eco-friendly system materials. So that's why I'm to review about these materials.
Okay.
Okay, Ms. Somratna, thank you for the nice presentation. Actually, this is not a question. So for my clarification, when you are doing this review, what are the your findings you find from the Sri Lankan situation? Are there any literature you have find from the Sri
I did
Lanka?
not find, no, madam, I did not find any studies from Sri Lanka. I covered all, location, I covered all worldwide.
but I did not find any research conducted in Sri Lankan situation.
But any other questions? Right. If there's no question, we'll move to the next presentation. Thank you very much, Ms. Somaratna. The title of the second presentation is Evaluating the Adoption of Climate Smart Agriculture Practices of Paddy Farmers in Cascade Irrigation Systems in Sri Lanka. Presenting author is Ms. H.M.N.B. Subasinghe. Over to you, Ms. Subasinghe.
Good morning, all. I'm Virendra Subasinghe from Faculty of Agriculture, Result University of Sri Lanka. I'm here to present my research on evaluating the adoption of climate smart agriculture practices of paddy farmers in cascade irrigation systems in Sri Lanka. This is the content of my presentation. First, let's move to the introduction. So, as we all know, climate change is a global challenge in agriculture because it mainly depends on climatic factors. Out of total population of Sri Lanka, almost half of the people are poor rural small-scale farmers. Majority of them live in dry and intermediate zone near the cascade. So negative effects that have been caused by climate change to agriculture will directly affect the social and economic well-being of those small-scale farmers. The Sri Lankan agriculture sector identifying the immediate need of CSA practices for the agriculture. So they have introduced a project on climate integrated water management project, which was initiated in Ghana and Intermediate zone of Sri Lanka. Main focus of that it was to introduce the CSA concept to improve the agriculture sector of Sri Lanka. This project has introduced different technological packages, including CSA practices for paddy. Even though many authorities have introduced different CSA practices, still there are a gap when considering the adoption of farmers. So, the main emphasis of my research to evaluate the adoption of climate smart agriculture practices of paddy farmers in cascade irrigation systems in Sri Lanka. These are the specific objectives of my research. In order to materials and methodology, my research have initiated in 5 districts as mentioned here. Then, the project have introduced different diversifying cropping models for them. As mentioned in here, they have introduced 3 models for them. So considering all of these, I have developed my sampling design representing 50 farmers from each cropping diversifying model. So the total sample size of my research is 150 farmers. In this project, they have introduced different CSA practices for paddy cultivation and different for OFC cultivation. When it comes to the data collection method, I have collected my data through structured questionnaires and focus group discussion. And for data analysis, I have used a descriptive analysis, Pearson method. And when categorizing responses, more than 80% is considered as highly adapted, 50 to 80% is considered as moderately adapted, and less than 50% is considered as low adaptable group. Now let's move to the results and discussion. So first, I have identified 4 CSA practices that have introduced for paddy cultivation and 5 CSA practices that have introduced for OFC cultivation. Then I assess the adoption level of farmers for CSA practices. So as mentioned here, in paddy cultivation, most of the farmers have adapted to downstream water management, which was 95%. Then less number of farmers adapted to parachute seed transplanting method, which was 4%. Then in OFC cultivation, most of the farmers adapted to soil conservation practices, which was 95%, and 30% have adapted to agronomic advanced services practices. Then I evaluate the average annual income level considering each of CSA practice. So in paddy cultivation, the average annual income level of farmers is high who adapted to agronomic advisory services. Even though most of the farmers have adapted to downstream water management practices, the average annual income of that is less than the farmers who are adapted to agronomic advisory services. Then in OFC cultivation, the average annual income level is high to practice soil conservation practices. Even though there are less number of farmers adapted to agronomic advisory services, their average annual income is also in a considerable level. Then I check the correlation between average annual income and CSA practices. In paddy cultivation, the Pearson correlation coefficient was 0.241, which indicated a positive linear relationship and indicate there is no strong or significant relationship between average annual income and CSA practices. In OFC cultivation, I got 0.921 coefficient between CSA and income. So there is a strong positive linear relationship between that two variables, which means there is a significant relationship between CSA practice adoption level and also average annual income level. Then I check the challenges and problems of farmers who adopted less for CSA practices. So according to them, there are key barriers for lack of knowledge, by animal attacks, in shopping on financial incentives, and reluctance to adopt new practices. So for conclusion, adoption level is varied from different CSA practices. Comparatively, less number of farmers adapted to CSA practices that have introduced organic cultivation, while considerable level of farmers have adapted to CSA practices that have introduced OFC cultivation. So according to the results, this study revealed that there is a positive correlation between CSA adoption and average annual income who practice OFC cultivation. So this recommend climate action plan with a holistic approach, which includes CSA practices, with considering the key barriers of farmers and also incorporating OFC for improving resiliency, diversifying crop system. So this is the end of my presentation. Thank you.
Right. Thank you, Ms. Subasing, for your nice presentation. Now the presentation is open for discussion. Any question from the audience or the panel members?
Ms. Subasinghe, thank you for your nice presentation. Do you have any suggestions to increase to more adaptation? What are your suggestions? How we can adapt them to the climate smart agriculture? Do you have any suggestions?
Am I audible, madam?
Now we can.
As I checked, most of the farmers that have less adapted to CSA practices, they say there are lack of some training, and also their awareness is less. And also they said that they have some wild animal attacks. So that is why some of the farmers not adapted to them. And also some farmers said that they are no, reluctant to adopt new practices, because they rely on the practices that they have practiced from the earlier, from their grandparents, like that. So I think it is better to have group discussion with those farmers who practice in traditional farming, and also think about their barriers, key barriers, because we have to go practical solution rather than theoretical things. So I think it is better to get a holistic approach, and also better to train them to cultivate OFC for paddy cultivation to improve their cropping intensity, and also as a resilient to climate change.
One more question. So that means they have knowledge, but they do not use these climate smart agriculture practices. Is that correct?
Some of them have knowledge, sir, but some of them said they are not adapted and they are not trained well.
That means the extension services or other things have the problems? Or did you check that things by evaluation or something like that? Did you evaluate that component, their knowledge or extension services or something like that?
In this project, sir, first they have give them training for all the CSA practices, but some farmers reluctant to go that practices, like to get that training sessions like that. That is why I think they are saying that they are not knowledgeable about CSA practices.
Okay, got it.
Right. I also have a simple question. What were the main reasons behind the varying levels of climate smart agriculture adaptation between the paddy and the OFC?
Because I think, sir, when come to agromet advisory service like practices, it is something that they are giving weather forecast to farmers.
So in there, they give a seasonally, monthly, and weekly rainfall pattern. Then they can get idea about what are the crops that they can cultivate according to the rainfall. And if we come to the paddy, they can if the rainfall is less, they can go to a 3.5 month paddy varieties like that. So if some farmer get good result from that agromet advisory service. So according to my results also, most of the farmers get average annual income level of those farmers who practice to agromet advisory services is high. So I think then farmers discuss about them. They are discuss about what are the practices that they can get high income. They only focus on high income. So I think that is the difference between adoption levels.
Okay. All right. Thank you, Ms. Subasinghe.
Thank you.
We'll move to the next presentation. The title of the next presentation is A Study on the Effects of Different Irrigation Water Hardness Levels on the Firmness of Gherkin during Vinegar Fermentation. Presenting author is Ms. Vijayasuri. Over to you, Ms. Vijayasuri.
I am Hashini Vijayasuriya from Faculty of Graduate Studies, University of Ruhuna. My research topic is a study on the effect of different irrigation water hardness levels on the firmness of gherkin during vinegar fermentation. This is the flow of the contents. Let's move to the background of the study. Gherkin, which belongs to family Cucurbitaceae, are widely cultivated worldwide, primarily for the pickle industry. A firm texture is essential in pickling cucumbers of any type to achieve high levels of consumer acceptance. During pickling process, fruit's texture can change due to the breakdown of pectic substances and osmotic effects. Various agronomic practices, including irrigation water quality, specifically water hardness, plays a significant role in determining the firmness of the fruit during pickling. In Sri Lanka, different areas exhibit varying levels of water quality, with water hardness being a significant factor. Identify the specific hardness ranges that negatively affect fruit firmness will help develop strategies to mitigate fruit quality during fermentation. In our problem statement, profit loss due to reduced firmness. There are several factors caused to them. Soil, nutrient management, climatic changes, stress and water quality. In this we focus on water quality, specifically irrigation water hardness. In different areas, different water hardness levels, then which range good for obtaining good firmness. Therefore, this study helps to identify suitable areas for gherkin cultivation. This is our objective, to evaluate the effect of different levels of irrigation water hardness on the firmness of gherkin during vinegar fermentation. In the methodology, we select open field located at the Faculty of Agriculture and in the planting material we use gherkin variety Kirti F1. Pots filled with soil was used as growing medium. In the harvesting stage, we harvested grade 1 gherkins and washed and put into vinegar solution. In the treatment preparations, first we dissolve 5 gram of calcium chloride and 2 gram of magnesium chloride in 100 milliliter of tap water and add a bowl solution based on the graph corresponding to treatments. Finally, we volume up to 1 liter. These are the treatments allocation according to the electrical conductivity. This is the experimental setup and the experimental layout. In the data collection, we collect data fruit firmness after 45 days using sensory evaluation and instrumental method. In the data analysis, we analyze data by one-way ANOVA using software and Kruskal-Wallis test. Mean comparison was done by using Duncan's multiple range test at a 5% probability level. Then move to the result and discussion part. In the texture analyzing using force gauge, T1 and T2 have a significantly higher values than the other treatments. In the texture analysis using sensor evaluation, T1, T2, T3, T4, T5 and T6 have the significantly higher value than other treatments. Therefore, we concluded that irrigation water hardness significantly affects gherkin firmness during vinegar fermentation. The highest firmness was observed in both instrumental and sensory analysis were recorded at 0.5 to 1 decisiemens per meter, where gherkins maintain higher firmness. In contrast, higher hardness levels, particularly 4 decisiemens per meter, resulted in reduced firmness. These findings emphasize that lower irrigation water hardness levels are optimal for producing firm pickled gherkin. As consumer preference leans toward firm pickles, adopting appropriate water hardness management strategies can directly contribute to product quality in the gherkin processing industry. This is the reference we used. Thank you for your attention.
Thank you, Ms. Vijay Surya. Now the presentation is open for discussion.
Thank you very much for your nice presentation, Ms. Vijay Surya. I have few questions. First one, according to your knowledge, so commonly, what is the normal hardness level of surface irrigation water that normally used in dry zone cultivation, and at the same time with respect to tap water or something like that? So normal hardness level?
Normally 0.5 to 3.5 decisiemens per meter. 0.5 to 3 decisiemens per meter.
So that is the range normally.
Normally.
Right. Okay. So with the increasement of the hardness, what are the other observation that you observed other than the firmness? Do you observe any other characteristic during the research?
In growth and yield also decrease.
Decrease.
Yes.
So you did not include those things in this part. So are there any other research or something like that?
Yes, sir. It is a research of other.
Right. So that is the case. So other thing, can you explain why you choose exactly that range of treatment 0.5 to 4? So you
normally range from 0.5 to 3.5. Because of that, we use this range.
Okay, so my concern was less than 0.5. So I do not choose that. So whatever, if it might have better yield or something like that, better firmness or something like that. So are there any other reason to not to choose less than 0.5? Okay. Right, thank you.
Any other questions? Okay, I have a simple question. Would similar effects of water hardness to be expected in other picked vegetables, or is this specific to gherkins?
I used only specific to gherkin.
Sorry?
I used specifically for gherkin because the pickle industry.
Okay. Thank you for your presentation, Ms. Vijay Surya. So we can move to the next presentation. The title of the presentation is Investigating the Impact of Smart Sprinkler Irrigation System over Manual Sprinkler System on Maize Crop Performance with Crop Model Scenario Analysis for Precision Water Management. Presenting author is Mr. D.M.R.M. Disanayake. Over to you, Mr. Disanayake.
Hello everyone, I am D.M.R.M. Disanayake. This is my research topic. Investigating the impact of smart sprinkler irrigation system or manual sprinkler system on maize crop performance with crop model scenario analysis for precision water management. The research mainly focused on maize crop because there is a high import cut from maize inside country and the productivity can be induced by improving agronomic practices, mainly irrigation, especially with using our technology. And the upstream-like crop models help to improve productivity while reducing the cost and time constraints in agronomic research. The experimental objectives were mainly twofold. One is calibrating the system and identification of drawback while comparing it with manual sprinkler system. And the other was using upstream scenario analysis, upstream crop models to find best irrigation scenarios. Turning into the material and method section, the field trial was done at Field Crop Research and Development Institute, Mahalupalana, with two treatments. Here, agronomic practices were followed by the Department of Agriculture recommendation and 8 growth parameters were measured during the experiment. If I explain about the initial calibration, zero value of the system calibrated at dry air condition and the 100 value was calibrated by immersing the sensors into the water. Then after the second calibration, system was automated between level 75 and 86. And these are the real-time moisture indicators, the TeamView app and mobile SMS. Here you can see the setup of irrigation control with indicator bulbs, knobs and buttons. In the other image you can see Arduino based circuit diagram of the system. The working principle can be presented by this figure. With the depletion of soil moisture levels to 75 level of the system, irrigation is started and when it comes to the 86 level, the process is going to be stopped. These are the upstream scenario analysis we used. The first scenario was based on the change in the irrigation amount and interval, and the second one based on the irrigation according to the growth stages, and with the third one was based on change in the irrigation amount and frequency. Here you can see the upstream scenario analysis dashboard. Moving into the result and discussion section. This calibration curve was developed using IoT system value and volumetric water content. These are the graphs showing the IoT value and volumetric water content fluctuation throughout the calibration period. With this curve identified that the value 75 represents the management allowable depletion level and 86 represents the field capacity. As a developmental requirements, there are especially the electrical isolation to prevent cross-plot dependency and sensor development with integrating machine learning algorithms approaches to system become very crucial. Compare the plant growth parameters and none of them didn't show a significant reduction. Considering about total water consumption and water productivity, they were high in manual sprinkler system, but the water productivity difference was not statistically significant. So here, the smart system has optimized water use without compromising plant health and yield. And especially this time period, there were huge rainfall events, and it is evident that this has been an effect to the IoT system responsiveness. These are the results taken from upstream scenario analysis. It was identified that applying 7.68 millimeters every 2 days, applying 80 millimeters at sowing, jointing and tasseling stages, and applying 90 millimeters 5 times within the growth period give best result. And here, well-maintained deficit irrigation showed best performance under favorable rainfall conditions at 3 growth stages, all around with 30 years weather data. And when we come to the conclusion, it is evident that the IoT system can match higher yields with reducing labor and also water usage while promising efficiency and sustainability. And for future studies, the lower and higher value of the system can be used as 75 and 86. As developmental requirements, it is important to develop soil-specific sensors with isolated connections and integrating machine learning algorithms. And with adequate rainfall at the key growth stages of maize, the best upstream irrigation scenarios are applying 7.68 mm for every 2 days, 8 mm at sowing, jointing and tasseling stages, and applying 90 mm 5 times. And this is end of my presentation. Thank you very much. And these are the key references I have used.
Right. Thank you, Mr. Disanak, for a nice presentation. Now the presentation is open for discussion. Any questions?
Thank you, Mr. Sanayaka. Thank you for the nice presentation. So you have doing that some modeling scenario. So this for my clarification. What are the drawbacks when you are then the constraint when we are practically use that smart sprinkler irrigation system, and do you especially concern about the cost component? Can you explain when compare the two methods? Do you have an idea?
Yeah, actually, if basically I explain about this, am I audible?
Yep.
This was done at Mahaluk Palama, but the season the research was conducted was impacted with high rainfall. That was one of the constraints I had to face. With that, sometimes the potential outcome cannot be implemented. That means potential outcome cannot be displayed very well. And other practical difficulties was actually these are the we used the low cost soil moisture sensors. Actually, they were designed with two brass probes and simple design for the cost effectiveness. And we were connected them to the IoT system control with wire connections, and they were disrupted by some rodents, and that is also a big problem. And there were other technical problems also with the cross-plot dependency and other temperature compensation were some drawbacks. And if I, yeah, definitely a cost analysis was also done. Here, if I consider about the manual sprinkler system for 1 hectare, we usually around 700,000 cost is we have to bear for the normal pipes and other things. For the smart system, the additional cost is we have to bear only for the IoT system controller. That means the controller, it is 60,000. It's so with the cost component, that is not a big. Actually, the system is designed for this level for some middle income farmers, not the lowest income farmers. So they can bear the cost. We assume that that's the answer.
Okay, thank you, Mr.
Okay, I have two questions.
Yeah.
Why you select AppSim specifically? Because there are many crop models. Why it's so specific?
Yeah. Actually, my research was mainly twofold. One was based on the IoT system, the other was based on AppSim analysis. So especially this AppSim model I use have been previously calibrated and validated for maize AppSim maize, this hybrid maize variety. I used the Pacific maize hybrid variety. It was previously calibrated and validated. So I have to use some other information, as you know, the other climate data, met files, and I ran different scenarios and get quick results.
Okay, so you did not perform the validation. You just used the calibrated model and apply it as it is.
Yeah, it was actually previously calibrated for this specifically for maize variety for in my Palm area. So
for someone else.
Yeah.
Okay, interesting. Right. Thank you, Mr. Disanayaka.
Okay, thank you.
We'll move to the next presentation. The title of the presentation is Development and Evaluation of Radial Weeder to Control Weeds Effectively in Tea Fields in Sri Lanka. Presenting author is Mr. AMWP Mathuranga. Over to you, Mr. Mathuranga.
My name is AMWP Mathuranga. Today I am going to present about development and evaluation of radial weeder controlling weeds effectively in tea fields in Sri Lanka. These are my contents. Let's move to introduction part. Sri Lankan tea industry faces significant challenge due to current economic crisis. Most of small-scale tea estate holders use manual weeding controls in order to reduce weeding cost. Development of radial weeder gives a solution problem related to manual weed control. These are aims and objectives. Let's move into research design and methods. This is overall experimental setup. In this research, start with data collection and field survey. In this field survey, we investigate about current weed control methods such as hand weeding, chemical weeding, and mechanical weeding, and we investigate about available machineries related to tea weeding, and that information used to development process. In development process, we first develop 2D model and 3D model. Then we tested the mechanism using test model and simulation. After confirming the mechanism, we fabricate the radial reader. Then we go to the evaluation process. In evaluation process, these parameters used to evaluate the radial reader, and it compare with hand weeding and chemical weeding. This is the radial weeder. This weeder use hand working mechanism. This weeder can perform slash weeding and scraper weeding using two blade attachments. This fork and main frame used to the connect to the different blades to the weeder. Foot pedal used to the push the weeder. Figure 3 shows major dimension of the weeder. This weeder use two kind of blade. This is first blade, slash weeding blade. This blade has upper blade and bottom blade. The bottom blade connect to the main frame and upper blade connect to the fork. This is second blade. It is a scraper blade attachment. It can perform scraper weeding. It can rotate 180 degrees and it connect to the fork. Let's look at result and discussion. This is field survey result. In this field survey, we find out grass is more prominent in small scale tea estates, other weed available at low frequencies. Hand weeding is more frequently used by small scale tea estate holders than mechanical and chemical weeding. Let's look at field capacity and forward speed. In here, hand weeding SW was significantly different from other treatment. When we consider about other treatment, slash weeding by radial weeder SBA, scraper weeding by radial weeder SCBA, and chemical weeding CW, there is no significant difference. Let's see, look at weeding efficiency. In weeding efficiency, there is no significant difference observed in chemical weeding, hand weeding, and scraper blade attachment. And however, weeding efficiency of slash blade attachment is significantly different from a chemical weeding. Let's we look at the weeding cost. In this weeding cost, our radial weeder had lowest weeding cost among treatments. This is key highlight. Next we look at surface soil disturbance. In surface soil disturbance, we observed highest surface soil disturbance in scraper weeding and hand weeding, lowest surface soil disturbance in slash weeding by radial weeder and chemical weeding. This figure shows how surface soil disturbance happening in hand weeding. Let's look at damage plant percentage. In damage plant percentage, there is no significant difference between treatments, and however, we observed somewhat higher amount of damage plant percentage in radial weeder because of its mechanical nature. Let's move to conclusion. Most of small scale estate holders use hand weeding than chemical and mechanical weeding. Radial weeder achieved above 80% of weeding efficiency in both weed control method. Considering the weeding cost, radial weeder had lowest weeding cost among treatments. This interchangeable blade make the difference. Considering the result of this study, radial weeder can be recommended to small scale estate holders as an alternative weed control method. These are the key references, and thank you so much.
Right. Thank you, Mr. Madhuranga. Now the presentation is open for discussion.
Yeah, thank you very much for your nice presentation, Mr. Madhuranga. So you have developed weeder, am I correct?
Yes.
Okay, so can you elaborate? So what is the major differences between conventional weeder and the one who you have developed?
Conventional weeding methods such as people use mostly hand weeding, and small people, some people use brush cutter, but its usage is very low. This weeder use a manual control mechanism, and we can perform two kind of weeding using interchangeable blades. That is key highlight of this weeder.
So when you are calculating the cost component, so you only consider about the labor cost, I guess. So what about the material cost and all those things? What are the materials you have used? And for example, so how
Yes.
much material cost need to be bear when you are preparing such a kind of equipment?
When I calculating material cost in chemical weeding, we use material cost also, and our development of this year is somewhat less production cost because of we use some uh, cheap materials for this weed.
Cheap material means, can you be specific? Can you be specific
Some
what kind of material you mean?
materials, GI pipes, some PVCs, like that.
So my concern about the damage to the soil. So you are mainly focusing on tea cultivation. So we all know about the soil erosion and all those problems are also related with the tea cultivation due to the slopy lands. So what is the effect of
Yes.
soil? Okay, soil surface. What is the effect of soil surface? Have you evaluated that? Or
When we using a scraper weeding in this field, there is a much soil loss. That's why we introduce slash weeding also. Slash weeding can affect to the reduce weeding cost, and that slash weeding method we can use to the some slopy tea lands, and some flat tea land we can use scraper weeding.
okay, one more question. So have you studied about the interval? I mean, so compared to the when we are focusing about the weeding, so we have to think about the first weeding and the time period for getting next weeding. So have you evaluated that thing? So I mean, so once you use the your equipment, so how many times that it need to take to go for the second weeding? For example, if we go for a manual weeding, it is take bit longer, right? It is persistent for a longer period of time since it is hand weeding. So when you use your equipment, what is the time period? So do you have the idea about that? Have you evaluated?
We not evaluated that.
Okay.
Normally, it's depend on the climate condition of the different areas. When we use scraper weeding, we can assume we can get more long intervals. When we use slash weeding, we need to go to the closer intervals,
Okay,
weeding intervals.
right. Thank you. So my suggestion is, if you are going this equipment for a further analysis, you better consider about these other parameters as well. So then you can go for a better evaluation, and that will be best for your equipment. Okay.
Okay,
Right.
thank you for
That's
this.
for that from me.
Okay, Mr. Maduranga, I have few things to clarify. So what is that, the width of the weed? What is the area normally removing the weeds? And also I have a concern about in which growth stage we can use this weeder for the tea cultivations. So normally the
This weeder can use
in which stage for
all growth stages.
every stage?
Yeah,
So
because
what is
this
the
yes.
What is the width normally that equipments, that area we needed to go facilitate to the weeding, machine weeding?
Normally the width is 261 millimeter. We can use half a circle, and this using this scraper blade, we can choose how much we do you.
So you know normally
weeding
the
and
total area is covered when that plant is growing. That there is no space
Yes.
and the total area is covered when that tea plant is growing. Can we use in that
Yes.
stage also?
Yes.
How?
This tested on the mature field, and we tested on the some younger field also, and we had good this positive result.
You have tested this for the slope land or the flat land? What is about the gradient you have tested, the land grade? It is okay for the every. I
Yes,
think these are my suggestions. You can better when you are going for the commercialization or the further development with the proper, if you can give the idea with the growth stage or every growth stage and what are the land slope land or the flat land likewise, and the other parameters also you need to consider land compaction. You have give the surface some data, but the root damage likewise things also we need to give the, you can further consider all the things for the further improvements. Thank you.
Right. Thank you. Since there were a lot of questions, I don't have any questions for you, Madhuranga. Thank you very much. We'll move to the next presentation. The title of the next presentation is Single Stage Chill Milling for Improved Energy Efficiency through Brittleness Enhancement. Presenting author is Ms. RWMKS Dilrukshi. Over to you, Ms. Dilrukshi.
I'm Sanjali. I'm from University of Colombo Institute for Agrotechnology and Rural Sciences. My research topic is single-stage chili milling for improved energy efficiency through brittleness enhancement. This is the content. Let's move to the introduction part. Chili milling, also known as size reduction, refers to the process of grinding or crushing chili into small particles. In conventional multi-stage chili processing, the material passes through several milling stages. The remaining 99% of the input energy was dissipated as heat, thereby as the temperature of the ground product. Additionally, single-stage chili milling is more cost-effective and environmental friendly. Objective: to analyze the effect of reduced moisture content on chili milling efficiency to determine the energy requirement. Let's move to the methodology. This study investigated the feasibility of single-stage chili milling as a replacement for the conventional multi-stage method at an industrial level. Using the standard sample of dry chili with an initial moisture content of nearly 12%, the chili's moisture content was reduced to 3.5 to 4% through storage in a dehumidified chamber. This pre-treated chili was then processed in a single stage using a pulverizer. The energy consumption calculate based on the mill motor current. For comparison, conventional chili powder production using the same chili without dehumidification involved the multi-stage process, pin mill, hammer mill, pulverizer. This figure indicates the experimental setup of the single-stage chili mill method and conventional multi-stage milling method. And various parameters were evaluated for the size reduction process of single-stage chili milling method and conventional multi-stage chili milling method based on energy requirement. Several modules such as Rittinger, Kick and Bonds have been developed to explain energy consumption during the grinding period. Statistically analyzed, the experiment were conducted in triplicate. All data obtained from experiment were statistically analyzed using the statistical package for SPSS software. Then move to the result and discussion. This figure indicates the comparison of energy requirement of single stage and conventional multi-stage chili milling method. There was a significant difference between the size reduction ratios achieved using the single-stage chili milling method and conventional multi-stage chili milling method. It is important to understand the reduction ratio when performing the process of grinding. Higher size reduction ratio indicates a fine particle size. Energy loss constant of the single stage are comparatively lower than conventional multi-stage milling. Grinding is an energy-intensive process. The efficiency increase with decrease in the moisture content of the material as brittle materials consume less energy during the grinding. Small portion of energy applied to the material used to loosen the bonds between the particles. The remaining 99% of the input energy was dissipated as heat, thereby increasing the temperature of the ground product. Energy consumption is relatively high in conventional multi-stage milling. And also specific energy of single stage chili milling is comparatively lower than conventional multi-stage milling. Specific energy is the amount of energy required to grind until mass of the feed material. The power requirements, particle size, and surface area are affected by several factors such as the initial size, material size, brittleness, and moisture content. Single stage chili milling method was found to be more energy efficient than the conventional multi stage chili milling method. Then move to the conclusion and recommendation. In conclusion, the study definitively demonstrated the superior energy efficiency of the single-stage chili milling over the conventional multi-stage milling method. The method used for reducing the raw material moisture content in the study was dehumidification using the heat pumps industrial viability that should be studied in detail. This is the references. My my presentation is over. Thank you.
Thank you, Ms. Dilrukshi, for the nice presentation. Now the presentation is open for discussion.
Thank you very much for your nice presentation, Ms. Dilrukshi. Can you elaborate what is mean by that single stage milling? Can you elaborate for my clarification? I'm asking.
Conventional, traditional chili milling performance, material passes through the several milling stages, pin mill, hammer mill, pulverizer. In this research, I used only pulverizer for the investigation of the chili milling.
Right. So that means that previous milling processes you skip.
Yes, sir.
All right. So what about the end material quality, the size of the particles and the end product quality? Have you observed? You observed the energy conservation. That is okay with me, right? So what about the end product quality? Have you observed this?
Sir, I also studied particle size distribution, bulk density, energy consumption. Energy consumption is part of my research.
Okay.
Particle size particle size of single stage chili milling is more fine. We can get more fine particles comparatively conventional multi-stage chili milling.
So that means that is a part of your research, I guess, right? I hope that is it okay to ask about those things, right? So my concern is that in terms of energy, that is okay according to this research. So the fine particle means what about the consumer preference? So is it more prefer than the conventional one, or what about that part?
Texture, we consider texture, uniformity coefficient, likewise.
Okay, so that means in terms of energy, this is better. So my question simply, can you tell me that in terms of consumer preference also, this one good or not? According
Good,
to your
sir.
knowledge? Okay, so that is the findings, right? Okay, so you have mentioned about that dehumidifier or something like that. That means to reduce the uh, moisture content surrounding environment. So how do you perform that part? For that part, do we need to bear some cost, or what is the method of doing that?
So, right, we used dehumidification for the two heat pumps. Chili pots stored in several days in the dehumidification chamber of the under the using the two heat pumps. We
So
reduce the moisture content until their absorption equilibrium moisture content.
right, okay. So that means you use energy again in this process for another separate activity. Is that correct?
Sorry, sir.
So that means during that single milling process, you use energy for dehumidifying activities as well.
Uh, we used energy for sir, we used energy using this heat pump dehumidification, sir. But we not consider that amounts for our calculations, sir.
Okay, so my concern, what is your thoughts? Don't you need to consider those energy consumption also? I
Consider.
guess. All right, okay. So anyhow, as my concern, this is good findings, but better to fine tune as much as possible for the better output. Okay, thank you. That is from me.
Any questions? Any other questions? Okay, I have a simple question. How does the reducing the moisture content of chili improve the milling efficiency and energy savings?
Sorry, sir.
How does the reducing of the moisture content of chili that improve the milling efficiency and the energy savings? What is the relationship?
So, we stored chili pots into dehumidification chamber using the two heat pumps and reduce the moisture contents through this process. Reduce the moisture content and increase in the brittleness. We can grind in more efficiently.
Right. Thank you, Ms. Dilrukshi, for the nice presentation. We'll move to the next presentation, which is the last presentation of this session. The title of the presentation is Assessing the Variability of Rainfall and Temperature in Major Paddy Cultivating Districts of Sri Lanka. Presenting author is Mr. M.D.A.N.H. Jayasundar. Over to you, Mr. Jayasundar.
For centuries, farmers in Sri Lanka have looked at the sky and planned their plantings. They just believed their visions. But what if the seasons forget their roles? What if the rains delay or arrive too early or don't come at all? So, or else, what if the sun burns too hot? So, very good morning to you all, and you all are welcome to my presentation. My name is Anja Sundara, and so today we are going to explore how this rainfall and temperature, the two main aspects of weather, are changing in four major paddy growing districts in Sri Lanka. So this is the content of my presentation. So according to the latest statistic, Sri Lanka is one of the top most vulnerable country to climate change. In recent years, we are experiencing high temperatures with significant variability in rainfall. So these extreme events mainly affect the agriculture of the country. So, as the rice is the staple food of Sri Lanka, I selected a major paddy producing districts to find out this variation of temperature and rainfall, which are the two main critical factors that determine the paddy productivity. So these are the objectives of my presentation. So this is the general objective and these are the specific objectives. And so when we come to the methodology, so this is my study area. So as the study area, I selected 4 major paddy producing districts, Anuradhapura, Kurunegala, Ampara and Polonnaruwa. And so as the data collection method, I get the sources from the Natural Resource Management Center to indicate or to represent the 4 districts. I selected 4 meteorological stations from each district. And I gathered data for 30 years. And in my methodology for the data analysis, I use several graphs to find out the variation of average annual rainfall, average monthly rainfall, and also average annual minimum and maximum temperatures with the standard deviation. So for the data analysis, I use the R software and I use 2 statistical methods called mean gain rule and sense log method to find out is there any variation. So this is the these are my observations. So according to the observations here, I get the variation of average annual rainfall. So here the highest annual mean annual rainfall is in Kurunegala and lowest mean annual rainfall is in Anuradhapura with their standard deviations. So here now we are coming to the variation of average monthly rainfall. So this graph shows the variation of average monthly rainfall of all 4 districts. So all 4 stations show the bimodal pattern, and from the Maha season is from September to February, which is the primary cultivation season, received the highest rainfall, while the Yala season from March to August has shows the highest deviation values. So this is the trend analysis result of rainfall. So we all know that according to the Menken-Dole test, we consider when the p-value is less than 0.05, there is a variation, and also when after that we did the sense slope analysis, and when we get a positive values for that, therefore the rainfall in Yala season in Anuradhapura and Polonnaruwa, there is a positive variation of the temperature, the rainfall values. And then we are coming to the observations of the temperature. So this is the graph that shows the variation of average annual, minimum and maximum temperature. So there, the highest mean annual maximum temperature was found in Polonnaruwa, and the lowest mean annual maximum temperature is in Ampara. And highest minimum mean annual temperature was found in Ampara, while lowest minimum mean annual temperature was found in Kurunegala. So then we are coming to the trend analysis results. So according to the Menken-Dole and sense log test, there is no tendency of variation in any districts for the minimum and maximum temperatures for both Yala and Maha seasons. So now we are coming to the conclusion. So Anuradhapura is the driest of the 4 districts, and it has the most unpredictable rainfall pattern, and also we saw that there is a bimodal pattern for all 4 districts, and from Maha season it has the highest total rainfall, and in the Yala season exhibited the interannual variability. And when it comes to temperature, the Polonnaruwa is climatically the most variable in terms of temperature fluctuations, and in Ampara is thermally buffered, and Kurunegala has the most stable temperatures. So, according to the results, we can conclude that there is a significant variation of temperature and rainfall. So, we should do more research and take more actions to find out more adaptation strategies, solutions, and to increase the productivity. So, ultimately, understanding variability is the first step, and next step is preparing for it intelligently, urgently, and collectively. So these are my references, and thank you very
Thank you, Ms. Jayasundara. Now the presentation is open for discussion.
Okay, thank you, Ms. Jayasundar, for this presentation and the findings. So you have find that there are in different districts, there are different unpredictable rainfall and the temperature. So, beneficial to adapt that because this is a normally timely important topics. Do you have any suggestions what farmers can adapt as a solution for this?
Am I audible to you?
Yeah.
Okay, so when I'm talking about my suggestions, the first thing is that the farmers, according to this, that as we can't predict the upcoming rainfall and the temperature and the climate variation, so I think that farmers can adapt to crop cultivations and rainwater harvesting, those ways that can use the water efficiently, the methods that can use water efficiently, and also agroforestry
Okay,
systems.
for the paddy cultivation, do you have any suggestions?
So when I'm talking about my paddy cultivations, we can use more drought and rainfall tolerant rice varieties, and the farmers can use more crops than rice and paddy, like crop cultivation methods they can use.
Thank you, Ms. Jayasundara.
Right, I have a couple of questions. According to my understanding, these 4 stations, they belongs to 2 climatic zones. Battalavad, I think it's in the intermediate zone, it's not in the dry zone. So did you observe any difference between these 2 zones, dry zone and intermediate zones?
Mostly, sir, when I'm talking about the yellow season, when I'm considering evaluating the results of the Kurunegala district and compared with the Anuradhapura district, there is more, the Kurunegala district has got more rainfall, as it is in the intermediate zone, compared to the other 3 districts. And also there is a variability of rainfall in the intermediate zone, like in Kurunegala district, than the other districts in the dry zone.
How about the temperature?
So when I'm talking about the temperature, there was no a very big changes, no variability, but there is a trend in increasing the temperatures. So mostly in the dry zone, the temperature has been increased when compared to the intermediate zone, like in Kurunagal.
I have one last question. So you check the temperature, minimum and maximum temperature separately. Is there any difference between the trend, the minimum and maximum, or the patterns or the slope?
Yeah, when I'm talking about maximum temperatures, there was no trend it is increasing more identifiable trend was not there. Like when I'm using the Mencken doll, there was no, the p-value was not less than 0.05 in every district. So I couldn't find any upcoming like trend in maximum temperature. But when I'm considering about the minimum temperature, like there were trend in Polonnaruwa and Anuradhapura districts. So like positive trend in minimum temperature.
Right. Thank you, Ms. Jacinda.
Thank you.
I think we are at the end of the session, technical session. Crop science and emerging technologies for sustainable agriculture. There were 7 presentations which covered diverse range from waste materials, climate smart agriculture, gherkins, modeling, weeders, chili milling, and the climate. I congratulate all the presenters for their presentations and successfully completion of the task. And thank you very much for inviting me to this session. And I would like to thank the panel members, Dr. Supun, as well as Ms. Katipearachi and Chaturi. Nice to see you too after a long time, Dr. Supun and Chaturi. And thank you very much for the opportunity. Right, I wish you all the best.
Okay, thank you, sir. Now we have come to the end of the last session of the first international symposium on agrotechnology and rural sciences, titled Crop Science and Emerging Technologies. Thank you, sir. Now we have come to the end of the last session of the first international symposium on agrotechnology and rural sciences, titled Crop Science and Emerging Technologies for Sustainable Agriculture. First, I would like to thank you, sir, Dr. Eranga Vimalasiri, for chairing the session. And I would like to thank our panel members, Dr. Supun Bandusekara and Mrs. Kasuni Keripiarachi, for sharing your valuable thoughts with us. At last, I would like to thank all the presenters who have shared their valuable findings with us. And also, I would like to congratulate them for disseminating their novel findings with us. And everyone, I'm inviting you all to stay tuned. Organizing committee will contact you. Thank you.
Good afternoon, everyone. Good afternoon, everyone. We have now successfully concluded all the technical sessions of the ISATRS 2025. We extend our heartiest thanks to all the participants who shared their valuable research and insight with us. The organizing committee ISATRS 2025 would like to share some update regarding your future activities. The symposium proceeding will be uploaded to the UCIARS website in near future. Your presenting author certificates and symposium kit will be sent to you via postal mail as you requested. For any updates regarding the dispatch of certificate and symposium kit, we kindly invite you to stay contact through your official WhatsApp groups. Once again, thank you for your active participation and valuable contribution to ISATRS 2025. We look forward to staying in touch and seeing you in near future.